And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless. And here it is, the much anticipated debate between myself and Erica on human evolution. Enjoy, because it's a good one.
Hey everybody, today we are debating human evolution, and we are starting right now. Ladies and gentlemen, thrilled to have you here for another epic debate. I'm your host, James Coons, and welcome to Modern Day Debate. This is going to be a fun one, folks. So we have two seasoned debaters with us, two very clever and quick on their feet type debaters. This is going to be a lot of fun, folks. So want to let you know, if it's your first time here, consider hitting that subscribe button, as we've got a lot more debates coming up. We are very excited. In fact, I got to show you just a few of these because they're going to be epic. So first, RN Ra will be facing off with Dr. McElroy. That's going to be a big one, and that's going to be in person. So we're actually going to be streaming that, live streaming it from Dallas on our little, you could say, mini debate tour. So that's going to be a big one. Then, also want to mention, Nathan Thompson and Team Skeptic will finally cross swords. So they are going to be debating a flat earth debate. That's going to be a lot of fun. That will also be in Dallas. Whew. Hopefully things don't get too crazy. It might, who knows? I don't know. You know, it's going to be, we hope it's not a fist fight. You know, we'll be all right. We're going to make it, folks. Next up, I want to mention, if you haven't seen this, where have you been? Have you been living in a cave under a rock? Michael Jones from Inspiring Philosophy and Matt Dillahunty will be facing off during our Texas debate tour as well. So, folks, I am pumped for that. It's going to be a lot of fun. That will be streamed live and in person as well. So, want to let you know, for today's epic debate, both of our speakers have their links in the description. So if you're listening and you're like, I like that, I want more, well, you can get more just by, it's like that little description box down there. You can find more right there. So, with that, dear goodness, I just realized, remind me, the format, if I remember, oh, that's right, I do remember now, thanks. It's a 10-minute flexible opening. So that means, like, we'll give them, like, a little leniency, but, like, once it gets up to 12, I will be so happy to cut Standing for Truth's mic. I mean, I will. It's going to feel terrific. So, I told him, I said, don't go over 12. Don't even think about it. So after that, we're going to jump right into open discussion, which I think most people, it's like a lot of fun there, followed by Q&A. So if you have a question, fire it into the live chat. I will pull those questions out of the live chat, and super chats go to the top of the list, and also allow you to make a comment during the question and answer if you'd like to do that. So with that, very excited, folks. We're going to jump right into this. And so thank you very much, Erica and Standing for Truth, for being here, first of all.
Oh, do we get to comment on that? I'm excited to be here. I'm ready to have a good time on this potential eve of World War III that Twitter's been going on and on about. So there's nowhere I'd rather be than here.
Glad you are here.
And that goes for me as well, James. I'm really looking forward to it. Love this channel. Everybody, please like and subscribe. Hopefully Erica and I can help make people's night a little bit better after the yes, like Erica said, the World War III scare. Hopefully not.
Yes, we hope everything is safe with that. And so it's been sort of a wild day, and we are glad to be here with you together. It kind of gives you the big picture. We've talked about this before. It's like we debate. Sometimes we even get a little snippy with each other. But at the end of the day, we agree on the big things, like hopefully making the world a better place, stuff like that. So with that, very excited to get the ball rolling. And if I remember right, Standing for Truth, are you getting the ball rolling, or?
Ooh, I think it might be me.
Erica, sorry about that, you guys.
Yes.
Long day.
Yes.
Fair enough.
No, please don't worry about it. I think it's because I'm the affirmative, but I'm not positive.
Deal. And I'm going to set the timer for, like I said, 10 minutes. I'll just let you know once you get up to 12 if you get there. And Erica, the floor is yours. Thanks for being here.
Okay, cool. So my name is Erica. I go by Gutsick Gibbon on YouTube, do a lot of videos on anthropology and bioanthropology and primates because, God, I love primates. I got a BSA in animal science, picked up a minor in biology and one in anthropology along the way, and I'm currently getting my master's degree, a master's of research in primatology, hence why I love the primates so much. So, human evolution, an undeniably wonderful journey. Now, from my standpoint, there is indeed so much support for human evolution across numerous fields that cramming it into a 10-ish minute opening statement is going to be kind of tough, but I am going to try to do it. Now, as I mentioned last time in my conversation with Kent Hovind, even Carlos Linnaeus, who was a creationist, admitted that humans are indeed primates because he recognized that we were eukaryotes, animals, chordates, vertebrates, mammals, eutherians, and so on and so forth down the line. And he also realized that similar to how this rhesus macaque here has its nostrils opening downward and how it has the dental formula of 2123 and shares 93% of our genetic similarity, we are both catarrhines. So the same reason that you would classify this rhesus macaque as a catarrhine, you would use to say, oh, well, I guess humans fit into that category as well. And I happen to find that very inspiring and cool. So we don't have to just use morphology, though. We can also have a conversation about paleontology, which happens to be a favorite of mine, because evolution can indeed make a prediction. If evolutionary theory is accurate and humans did indeed evolve from an ape-like ancestor, which we would currently in the field consider Sahelanthropus chadensis, then we can say that if we share a common ancestor with chimpanzees, we should see the emergence of human morphology through geologic time. And paleontology displaced this gradient from our ape-like common ancestor with chimpanzees, Sahelanthropus chadensis, to modern humans. So essentially what we're saying here is if this is indeed the case, we should see slowly through time the appearance of novel human traits. And with this comes the macroevolutionary concept of speciation. So while B may not be able to interbreed with E, B may have been able to interbreed with C. And through this concept of small, novel microevolutionary changes, if you like, we do indeed see evolution occur. But of course, that is only the case if we are seeing this massive change occur through geologic time. And that is indeed the question. I just threw this one. Forgot I had this one in here. I love Australopithecus africanus almost as much as I love Afarensis, which would be Lucy. But as I asked Kent in the last debate, if you were to pick one of these, he likes to do they have a 5-year-old come up and tell us which one is the difference in order to differentiate biblical kinds. If you were to do that here, you'd be kicking the chimpanzee out and you'd be keeping Australopithecus africanus right up here in the mix. And of course, it has a myriad of lovely mosaic traits from the semi-parabolic palate, that is to say the shape of the upper palate, ventral frame and magnum that allowed it to stand indeed bipedally, which I do hope we have that conversation today because I think it's quite a good one. Sort of some retained neoteny that's continuing, and a pelvis that is, of course, looking very human-like. But like I said, can we prove that all these organisms, according to the evolutionary model, did indeed live at slightly staggered different times over the course of millions of years and not all at the same time and then perished in a global flood? Yes, we can indeed do so, thanks to the radioactive decay law. Now, the radioactive decay law is essentially this process of spontaneous transformation, essentially decay, rather, of a parent isotope into daughter isotopes. And when we look at these minerals that we find fossils in, we can get what's called an absolute date by looking at this ratio. Now, the radiometric decay law, radioactive decay law, is indeed a firm law in physics. It additionally covers the nature of decay constants and half-lives and indicates that to our current knowledge, decay cannot change in any meaningful ways in nature. Because frequently, what creationists tend to do is they say, oh, well, maybe it worked differently in the past, or maybe the flood accelerated this radioactive decay and gave us these dates that seem to say millions of years. But there's a problem with that. And that problem is when things decay, they release a ton of heat. Now, we're going to talk about that in just a second. But the RATE group, which is a young Earth creationism group, they were discontinued back in 2005. They spent a ton of time trying to prove that you could indeed accelerate this without problems, but they couldn't. And this is a quote essentially from that report. They admitted that a young Earth position cannot be reconciled with the scientific data without assuming exotic solutions will be discovered in the future. No known thermodynamic process could account for the required rate of heat removal, nor is there any known way to protect organisms from the radiation damage. And interestingly enough, a Tufts geologist went ahead and decided, all right, let's see what happens if we try to cram all the heat that results from accelerated decay from a 4.8 billion year old Earth to a 6,000-year-old one. And it's not a good situation, folks. He essentially found that at the time of Adam and Eve, if you're cramming all of that heat release into a 6,000-year time scale, the surface of the Earth is going to be 70,000 degrees Celsius for every square kilometer. And at the time of Jesus' birth, assuming a generous geothermal gradient, we're going to see around 400 degrees Celsius for every square kilometer. There's a link at the bottom there, and I can give to James if you'd like. But essentially, as far as we know, radiometric dating works because there's no reason that it shouldn't. There's no empirical question that has essentially been raised to say, well, what if it did work differently? It's rather firm. But of course, genetics are some of the more interesting and novel supports for human evolution that have popped up recently, because when you align our genomes side by side with the members of genus Pan, that would be the chimpanzees and the bonobos, you get a 98.8% similarity when you're coding for, or when you're comparing coding base pairs, which is fascinating and very cool, because when you compare their genome to the rest of the apes, you find that they are actually our closest living relatives just as much as we are theirs. They share more in common genetically with us than they do with a lowland gorilla, which is really cool. Now, I said this to Kent as well, but essentially that genomic comparison I just mentioned is just a souped up version of a paternity test. So the key thing to take home here is that if relatedness is determined by genetics in humans, why isn't it the same across species? But we can also look to the chromosome 2 fusion. So human chromosome 2 is a fusion of 2 chromosomes found in the other great apes. How do we know this? Well, this is a little picture of a chromosome, and it's tipped with telomeres. Now, the cool thing about that is telomeres have a very specific identifying sequence, which is TTAGGG, and its reverse, which would be AATCCC. They also have centromeres in the middle. So essentially, if you are accepting of evolution, you're going to make a prediction that human chromosome 2 is a fusion of these 2 hominoid chromosomes, 2A and 2B, sometimes 12 and 13 is what they're referred to as. So we should find telomeres in the middle of human chromosome 2, because if they're fusing right, they should fit together like that, and you should find endpoints in the center of our chromosome, with the upper portions of that chromosome being nearly identical to those separate chromosomes in the apes. So is that what we find? Yes, it is. It's very cool. So the banding in human chromosome 2 matches almost precisely to the banding that chimpanzee chromosomes 12 and 13, 2A and 2B, like I mentioned earlier. And then you have this inactive centromere in the center. Interestingly enough, and I do hope we get to talk about this as well, but this centromere is known to be a centromere, even if it may have a functioning gene on top of it, because it does indeed contain that TTAGGG sequence that I just mentioned. So this is a prediction that evolutionists made that did indeed come true back in 1991. This is the title of the paper. It's a good paper, it's a little lengthy. So we could also talk about the gulo breakage. So the gulo is essentially a broken gene that in most mammals allows them to synthesize ascorbic acid so that they can make vitamin C and they don't have to eat it, whereas we have to eat our vitamin C. Essentially, this would have broken around 61 million years ago with the omomyids and the adapids, which are little almost tarsier and shrew and lemur-like guys. This means that, like I said, we can't synthesize our own vitamin C. But the interesting thing is this breakage is in the same location in every single primate that is a haplorhine and onward. And furthermore, while gulo breaks also occur in other mammals, it's only in the primates that we get the CAMP gene expression by vitamin D. So what creationists need to invoke here is a separate occurrence of this breakage in every single primate lineage, as well as a separate CAMP alteration. That's a hard sell for me. We could also talk about the trouble with tools. So I didn't bring this up last time, but I think it's quite an interesting one. So essentially, there's trillions of tools strewn all across the African continent. They're typically these napped tools that we see, like Oldowan and Oldowan style tools that you get by taking 2 rocks and kind of chipping them together in like the classic caveman sequence. And there's no natural way that these tools can form. You can look at a cool picture of them. You're not going to get this by rolling a rock down the hill. But we have 15 to 150 trillion with the T of these guys laying all across the African continent, based off of an estimation of 250,000 stone tools found in approximately, per square kilometer in Libya, which is a whole lot of tools. So yes, it's a bit extrapolated, but let's see, 250,000, that's quite a few in and of itself. So the question is, how do creationists deal with all these tools? Because under an evolutionary lens, right, we've got several million years for organisms like Kenyanthropus platyops or Homo habilis to make all these tools and leave them all around as they migrate across the continent. So it's basically a numbers game, because the young Earth creationists kind of hold, generally speaking, at least Answers in Genesis does, where I got this lovely timeline, that there's a Stone Age that occurred approximately 500 years maximum. Yeah. And this would be essentially when everybody left the Tower of Babel. So you've got 500 years, you've got 25 years per generation with a replacement rate for the population, 10,000 artifacts produced by each person in their lifetime, and an average population size of about 100,000. Can you get all these tools with those population rates given by Answers in Genesis? And the answer is no, you can't. It's very problematic. So young Earth creationists essentially have to have a small band of people leave the Tower of Babel in 2250 BCE, migrate to Africa, forget all of the technology they once had in Mesopotamia, learn how to fashion rocks into tools, which is a technology in and of itself, and extract 40 million pyramids worth of rocks from all over Africa, fashion them into 15 trillion, and that is the lower end of stone tools, before placing them all across Africa in approximately 150 to 500 years. To steal from Kent here, that's imagination stuff. It's not going to happen. So essentially, to prove human evolution wrong in this context here, you're going to have to invalidate genetics by relatedness and genetic similarities in human genus Pan across their genomes, the human chromosome 2, and the Gulo break. We're also going to have to produce a mechanism for accelerated decay that solves the heat problem, which I think is going to be somewhat difficult. We're also going to have to introduce morphologic means of separating the humans from the rest of the apes. And we're going to have to produce a mechanism for those tools to form naturally, or a proper population growth that is going to account for all of them in such a small time frame. So I'm going to cede the rest of my time because I'd like to get to the discussion.
Thank you very much, Erica. Next, we'll go over to Standing for Truth for his opening statement. And Standing for Truth, I have got the clock set, and the floor is yours. Thanks for being here. Bet you have yourself on mute. Whoa, wait a minute. I think. Oh, no, okay, full screen. Okay.
There it is.
I thought I lost you. Okay.
Let me move this to the side. Okay, let me start my timer as well, just so I know. Let's see here. I got it set max 12 minutes. Okay, let's go. Okay, thanks so much for your opening there, Erica. I'm going to try and cover as much as I can. I've got my opening here. I'll address a few things that she talked about earlier. For the decay rate, according to catastrophic plate tectonics, if rocks were exposed to heat and pressure, this could speed it up. Heat from below the crust coming up to the ocean, then to continents, of course. So volcanic activity hitting rocks worldwide. She talked about human chromosome 2 fusion. Well, that purported fusion site's actually a functional DNA element in a human gene, so that's been overturned. When it comes to the human-ape similarity, they say it's about 99%, maybe 98%. New incoming data suggests that it's more like 88% similar. That's 400 million DNA differences that separate apes and humans. Also, the chimp genome is 12% larger than humans, so that helps separate us from chimps. They ignore things like copy number variations, and of course, there's huge gaps on human and chimp DNA. So let's get into it here. Debate human evolution. Looking forward to this one. Let me see. Okay, can you see my slides?
You can see them.
So she talked a lot about nested hierarchies as well. I'll get into that. There exists, though, an unstoppable crisis for those that believe apes, humans, and banana plants are related through common ancestry. Now, this crisis is that of genomic degeneration. The evidence is absolutely overwhelming that mutations are accumulating. I'd like to ask Erica to provide us with a type of selection or mechanism that can rid the population of so many deleterious mutations that are mostly unselectable and invisible to selection. So unfortunately for human evolution, which is the topic of this debate, both ape and human genomes are degenerating due to this mutation accumulation. This evidence is extremely strong. This strongly indicates that neither ape nor man could have evolved. They both must degenerate. In fact, many of the hominin fossils here that Erica uses to prove ape to man evolution clearly display anomalous pathologies that indicate that in the past, several isolated and inbred human populations have experienced reductive evolution. This goes for Homo neanderthal, Homo erectus, Homo naledi, the hobbits, of course. There is no progression. This type of reductive evolution has resulted in genetic pathologies, reduced body size, reduced brain size, and as a consequence, tribal extinction. This is expected given a biblical-based model of post-Babel conditions and post-Babel dispersion. Genetic degeneration is a very real phenomenon occurring in living populations. If what we observe today in the present is reverse evolution and not forward evolution, the same thing would have happened in the past. And it is very well documented that what we truly observe in the hominin fossil record is genetic degeneration. For example, numerous of the bones of the alleged Homo species like Hobbit, Erectus, Naledi, Neanderthal, the ones that she used here in her opening, appear to demonstrate testimony of severe inbreeding and genetic decline. Therefore, many of the troubling human fossils that Erica used in her opening presentation are much better understood to be devolution rather than evolution. The so-called primitive features that Erica here points to as a progression from ape to man is actually genetic degeneration. They weren't pre-human tribes. More accurately, they were human tribes that were persistently inbred and were in genetic decline. We can see here a paper actually that the claimed primitive humans are inbred subpopulations. This was a paper by Eric Trinkos. You can see it here. He's a paleo expert, and he noted that a total of 75 documented developmental abnormalities from 66 individuals spanning the Pleistocene. The author also explains that the remarkably high frequency of skeletal pathologies could not possibly be due to chance. This is exactly what we'd expect based on a post-Babel dispersion of isolation and subsequent inbreeding. The author suggests that these populations were almost certainly undergoing genetic degeneration due to inbreeding, as is expected in any small and isolated population. So these human variants, they all lived in isolation, and given the situations associated with each variant, the specific settings would lead to these founder effects, isolation and inbreeding. And what's funny is the author obviously doesn't conclude the biblical-based model, but these findings and more strongly support the biblical-based model of degeneration of small subpopulations, which is exactly what our model talks about. We got here just recently, I believe it was in 2017. This negates all her dating methods because we've got fossil footprints, anatomically modern human-looking footprints that debunks human evolution in the same layers or lower than the Australopithecines. So anatomically modern human-looking footprints have been discovered here in Crete. I believe it's in Greece. They date approximately to 5.7 million years old. So these findings here are clear evidence that Lucy, Ardipithecus, Australopithecus sediba, Homo habilis, etc., they're not our evolutionary ancestors, as Erica promotes here. And the reason is because these footprints indicate that creatures with human feet lived much earlier than these so-called human ancestors. So Erica's going to have to answer this question if she wants to look to rocks. It's genetics, genes, traits. These are what are inherited sperm and egg, not a rock, not geology, not geography, but genetics. So she's going to have to explain these anomalies to her evolutionary theory. I think this article here on Evolution News puts it perfectly. They say, when the oldest known evidence for hominin feet predates the alleged African ancestors such as Artie and Lucy, but already shows relatively modern human footprints, what is more congruent with this new evidence when looked at without bias? A gradual Darwinian evolution, or rather a saltational origin that requires intelligent design? Just a few points here. Evidence from paleontology, archaeology, and modern genetics all irrefutably confirm that Neanderthals were in every way fully human. Erectus, for example, was able to sail, which clearly speaks of human intelligence. Homo floresiensis, which Erica helped me say in the pre-show, thank you so much, was also very clearly human, as indicated by their impressive cultural inventory, ability to sail, endocast scans that reveal a modern human brain, an anatomy that is modern human looking. Reduced brain sizes, for example, can be beneficial in certain conditions, such as starvation. This explains the smaller brain sizes that we do see in some of the hominins. This is because brains are energetically costly. This is all evolution in reverse, and these pathologies and variants can occur quickly in isolated populations. This is all expected and predicted in the biblical model post-Babel and post-flood. Nested hierarchies she spent some time on. Before we even had DNA sequencing technology, it's quite obvious at the physiological level, humans and the great apes had a lot in common in terms of appearance, basic body structure. Both mammals have binocular vision, etc. There's some major differences, of course, though. Just look at the feet of the great apes and the shape of their feet. They have feet that look like hands. They use these for grasping purposes, of course, swinging from trees, etc. Now, humans have feet, on the other hand, that are built for upright walking. We are similar in these areas, and therefore we should have similarities genetically as well. This should not surprise anybody, and this is in no way, shape, or form proof that humans, apes, and pine trees are related. Now, of course, evolutionists like to look at these similarities and imagine that all life forms are related, but this is not empirical. This is actually in the realm of fairy tale. The bigger question we are trying to answer is, why are humans and great apes so similar? It doesn't matter whether we look at the anatomy, the physiology, the genetics. They all tell us that we are a lot more closer to each other than pine trees and dogs. Now, evolutionists will say the similarity genetically is the high 90s, like Erica was saying. It's just false. It's more like 88%. But regardless, we are pretty close. What does this tell us about our origins? The evolutionist seems to think to themselves that if we really are made in the image of God, shouldn't we as humans be distinct from the rest of biology? Well, maybe in a way, yes, but in another way, no. The Bible doesn't imply in any way that we have biological life and then human, which has absolutely no similarity to any other species out there. There is no reason for us to believe that based on a biblical model, there should be no type of hierarchy in the biological world. There's no reason to believe that we shouldn't be more similar to one species, for example, the apes, than to another species, for example, a wolf. Naturally, we as humans have designed things with groups within groups patterns. If you were to look at the types of vehicles or modes of transportation humans have created, they naturally form groups within groups patterns. A sedan has a tremendous amount of similarities with other sedans. Now, a semi-tractor trailer is obviously much different than a sedan or a car, but these two modes of transportation are similar enough to each other that it sets them apart from airplanes. This is the groups within groups patterns that emerges when we look at all the modes of vehicles or transportation that humans have designed. This is an empirical observation, and this is something we naturally do. It appears that God created kinds in the beginning that are different from us as humans, as well as kinds of creatures that are more similar to us. This is a reason why Aaron Ra's phylogeny challenge is fallacious. We too predict nested hierarchies, morphology, anatomically, and genetically. Evolutionists miss the bigger picture. Yes, we can find hierarchies in all of biological life, including humans and apes, but apes don't build airplanes, apes don't write books, apes don't compose symphonies. Chimpanzees and other animals don't do any of these things. Mankind has built the space shuttle and has accomplished wonders. So yes, there are groups within groups patterns in all of life, but this is simply a hallmark of design. The fact that there exist these natural and nested hierarchies is exactly what we'd expect if God created the world. We can see here, even if we got into transitional forms, we create things that blend the features of two different types of vehicles, amphibious assault vehicles, for example. We can see here that all the evidence regarding mitochondrial DNA, Y chromosome, the low genetic diversity that we see all points to Adam and Eve. Evolutionists need to ask themselves, if they have an open mind and they were to look for evidence for Adam and Eve, what would they expect to find? Because all human beings are profoundly similar genetically, and this points directly to creation. I'd like to talk to Erica here about the evolutionary out of Africa bottleneck and how that may be feasible.
1 minute.
I've got 1 minute here. Therefore, going into the discussion, a few questions that I would like to ask her are: the out of Africa scenario, due to the resulting inbreeding of a population bottleneck of 10,000, how is this even remotely feasible? I'd like to ask her about the Y chromosome of apes and man and how they're so dissimilar, less than 70%. The Y chromosome should be the most similar. So I want to ask her that question, as well as genetic degeneration. These near neutral deleterious mutations are accumulating. What type of selection can stop this genomic degeneration? I've got a lot more here, but for the sake of time, we'll save some of that for the discussion. Thanks so much.
You got it. Thank you very much, Standing for Truth. We will now shift back to the dialogue boxes and let her rip. So, thanks for your questions so far, folks. A lot of great questions. Excited to ask those. And now we'll go into open discussion. So, the floor is yours.
Okay, well, there's definitely a lot to talk about. First, I would like to quickly say they have actually tried time and time again to speed up or to accelerate decay with heat and pressure. And the most that they were able to do was 1.5%, which I believe it was Woodmarap, who's a young Earth creationist, commented on it and said, yeah, that's nowhere near good enough. We're going to have to, invoke some exotic solutions here. So, I mean, it's easy to say, yeah, we could just have a lot of heat and a lot of pressure, but that's never been demonstrated in the slightest to be good enough to get it anywhere close to what you need, especially because that 1.5% increase of decay wasn't even in a geologically relevant element. But more specifically, I would like to talk about...
If I could just respond to that real quick.
Yeah, sorry, go ahead.
No problem. I do think that the entire, radioisotope dating, the methods, for example, they do have unprovable and unknowable assumptions, right? For example, are the initial conditions of the rocks known? Clearly they aren't. So geologists, they weren't there when the rocks form. So at the end of the day, they don't know, right? For example, how do they know how many uranium atoms, if they had no lead atoms, for example? But I think it comes down to the fact that we do find now these anatomically modern looking human footprints in layers, either with the Australopithecines or lower. Therefore, how can there be modern human looking footprints in layers with our so-called Australopithecine ancestors?
Yes, so I would love to talk about that Crete footprint because I have looked quite extensively into this. I had this question asked of me about a year and a half ago on the Debate Evolution subreddit. But to quickly kind of take a step back and make kind of a comment on the radiometric decay thing. Yeah, we kind of do know the starting ratio because we do have rocks that do indeed form today. I see no reason why, since we've literally never observed a rock forming that didn't have a complete, parent favored ratio of isotopes, why we should expect that back then it was different, especially because, and of course, I'm sure we can have a conversation about mutation rates. There are reasons why we do say, well, some rates are constant and some are not. But there is, of the hundreds upon hundreds of thousands of times that we've radiometrically dated things and observed new rocks form, we've never been given any reason to suggest that ratio wouldn't be in favor of the parent, and the physics inform that. But I probably shouldn't have included SFT because this is a conversation about human evolution. It's kind of my bad. I guess that was a little.
Well, no problem. I definitely don't like to leave anything hanging because you did.
Sure.
Again. I just want to say, for argument's sake, I mean, I do stand behind the fact that it's genetics, it's traits, it's our genes that are inherited sperm and egg, not a fossil, not a rock. So, I think we should definitely look to genetics, morphology, anatomy in regards to human evolution. But because of the heat, I do want to say, based on our model of catastrophic plate tectonics, right? Subduction friction, for example, with the continent will make heat and make the rock more plastic. So indeed, this subduction can happen rapidly. This is actually called runaway subduction, or speed up the subduction, which can explain the necessary speeding up of the decay rate. But in regards to the hominins, which I do want to focus on, I'll let you comment on the footprints in Crete as well. But as far as I know, the dating methods commonly used to date the hominins, they do yield extremely inconsistent ages. Therefore, the paleoanthropologists seem to only use the dates that fit their story, right? So the dates that conflict with the proposed ape to man timeline are routinely rejected. Like, I'm guessing that these anatomically modern looking human footprints, the dates are probably going to have to be changed now, right? Because they've done it so many times before. These unacceptable dates are routinely assumed to be corrupted by sample contamination or whatever reason that they have, even if they have no evidence for said contamination. So it just seems like selective use of data. And you mentioned the genetics and the mutation rates. Well, I mean, we have a molecular clock regarding the Y chromosome and the mitochondrial DNA, which actually brings us back to 6,000 years. So, I mean, if you want to talk about dating, we can look at traits, genes, and genetics.
Yes,
that gives us a date of 6,000 years. Go ahead, Erica.
Well, I'm totally with you, but that, I guess I kind of opened Pandora's box there because there was a lot of points. So I would very quickly, I'm going to make a comment on Crete, on that Crete footprint. Have you seen a picture of it? Because I have. Did you look up a picture of it?
Yes.
Yeah, it's not very footprint-esque, especially considering what trackways tend to produce, especially given the sediment that it's kind of placed in. It's very unlikely, at least amongst the professionals that I have spoken with, that is actually a footprint at all, specifically because it's a single print. It's not, there's only one print that's being proposed. It's not a trackway like the Laetoli footprints where you've got, dozens of footprints with where you can actually take a plaster cast and get the balls of the feet and the toes and the heel, and you can actually see how the individuals that made those tracks bear their weight. You can't do that with the one in Crete. And
right.
I tend to think, if you go out and you pour a bunch of water in the mud and then you walk through it, you're not going to get one footprint. So I actually, everything I have seen on the Crete print suggests that it is not a print at all. And at least, and moreover too, one print, let's say you could prove that it was a print of something, right? That wouldn't overturn the entire timeline that we have now, particularly because we know for a fact that Europe used to have a booming population of primates. It would indeed be entirely possible that you would have apes living in the Oligocene and the Eocene.
Isn't it true that these footprints were found outside of Africa? And are you saying there was only one set of footprints that were found? And because from the article that I had posted in the opening presentation, the headline does say ancient footprints in Crete challenges theory of evolution, but what actually made them? I've looked at the source material as well, and I didn't find any data suggesting that they weren't actually footprints. I saw some paleoanthropologists or some scientists in the field claiming that maybe it was some type of extinct gorilla that had a footprint very similar to modern humans. But I didn't see anything about what you're claiming. So I'll definitely look into that. But if they are footprints, especially if they're outside of Africa, I think they're near Greece.
Yeah, they are.
That could challenge the theory.
It would add a lot of nuance to it if they were indeed footprints. But again, I would heavily suggest looking into it because I read the paper and then I looked for commentary on the paper, and then I also asked a past professor of mine kind of what his thoughts were. And everything that I've read is suggesting, one, it's very likely not not prints, because all I could find was, as far as pictures go, was a single kind of imprintation in the sediment that does, first of all, it doesn't particularly look like it would be in the shape of what you're kind of proposing as an anatomically modern human print anyways.
Are you saying that it looks totally...
Hold on. Oh, forgive me. Stanford, just to be sure that because there's a lot of info going back and forth, I want to be sure that she gets plenty of time to respond.
Of course. Go ahead, Erica. Take your time.
Yeah,
I just
sure.
want to say one last thing about the footprints when you're done.
Yeah. I mean, my point is that it's not, even if it were a footprint, which there's very, very few people standing up and saying that it is. And even if it were a footprint, there's even fewer. I don't know of anybody saying this is a hominid, or rather, a hominid. It's the wrong shape if you're trying to propose that this is anatomically modern. That's all I wanted to say about that. I don't find it very compelling.
If you're done, I definitely want to see. I'd want to see the paper or the source that says that they're not really footprints, because I've looked into it thoroughly and I never seen anything of that. So I'll definitely reserve judgment until I see that paper. But you did mention Mary Leakey, which about 25 years ago discovered some footprints, which are in all the source material that I looked into and everything that the paleoanthropologists described it as being indistinguishable from modern human footprints. So
100%. I have got to get it on this, because I had a conversation. I used to get coffee with a young Earth creationist friend of mine who was a coworker, and we had a very lengthy conversation about the Laetoli footprint. So I am on top of this.
Well, before you explain it,
sorry. Go ahead.
Okay, so you would. So you would agree that they do look exactly like modern human footprints, and they were what? Were these discovered or dated at about 3.5 million years ago in actually where were they
They're in
discovered?
Tanzania. They were found in Tanzania. Though
Okay.
they're the Laetoli footprints. So and
Right, the late
I
Tolly
would
footprint.
yes. So as far as them being indistinguishable from humans, I'm going to tell you about a very cool study that you should look into because I found it fascinating. Yes, they do look very similar to an organism that has an inline big toe. Now, if you look at the very like this is the difference between these and the Crete tracks because you can see the individual toes on some of these prints are incredible. You'll see that no, the toe isn't completely in line. It's just very, very close to being in line. So what the researchers of that project of this paper that I'm about to tell you about did was they were like, all right, well, let's take casts of it and then let's see if we can figure out how whatever was making these prints bore its weight. Because essentially, as I'm sure you know, the biomechanics behind walking on 2 feet are very, very specific. It's, there you have to have, proper weight bearing on the knees and you have to have a specific arch in the foot, which is great because you can tell from prints how something bore its weight. So what they did was they had a bunch of hunter-gatherer individuals from Africa. Someone similar to like the Hadzabe. They had a couple dozen of them walk in soft sediment, and then they took a cast. Then they had some chimpanzees walk across soft sediment, and then they took a cast. And then they took the Laetoli footprints. And did you know that the individual that made the Laetoli footprints bore its weight in a perfectly intermediate fashion between the two? And the researchers who published this paper said it is absolutely certain that whatever made these tracks was not a human and it was not a chimpanzee. And they're not even certain, I believe, that it was Afarensis. It could have been a.
Well, did Mary Leakey? I'm sorry. I just because I'm trying to follow you. Mary
Sure.
Leakey, the one that discovered these footprints 25 years ago, and she admitted that they look nearly identical to modern human footprints. I understand you're saying that there's a few differences that suggest otherwise. Did she conclude that it was from some type of hominin, or did she conclude that it's from some type of Australopithecine? Like, what was her conclusion on it?
She concluded that they were from Afarensis because they have Afarensis feet, and the toe is close to being as in line as an anatomically modern human.
I thought that was Donald Johanson that concluded that, because I know that he concluded that all these different bones were.
Well,
just to be sure that in case she wasn't finished.
Yeah, sure. Well, thank you, James. But I'm sorry. I do tend to go on monologues, so feel free to jump in if I kind of get going. I just get really excited about this stuff.
I definitely like to see, the data on that. It's my understanding that Mary Leakey did disagree with Donald Johanson and him concluding that all, the footprints, all the different bones that were discovered in both her area and his area were actually one species. I do want to, because we were talking about the trackways for a while, I did want to ask you though, regarding, because if we're going to talk about human evolution, right? I understand that you would believe in, actually, would you believe in the out of Africa theory, or would you hold to more the multi-regional theory in regards to Homo erectus?
I think it's kind of important that we wrap up that past conversation because yes, Mary Leakey and Johanson had different opinions, but the species, as if I remember correctly, and as far as the people go, I can get kind of hazy. There are a lot of people involved in the community, but at least as far as I remember, the dispute was not between whether it was Afarensis or a modern human, but whether it was Australopithecus afarensis or Australopithecus anamensis, who was also in the location and has also been at least purported to have, I don't think we have the feet of anamensis in the same, we have a lot better representation of Africanus and afarensis than we do anamensis. But essentially the dispute was not, I feel like that's a bit of a mischaracterization, right? Because it's not like Leakey was saying, oh, they're so close to human, they're definitely human. She was just noting how incredible it was because the dispute that was going on at the time in the anthropological community was whether or not our ancestors, at least according to evolutionary theory, got big brains first or started walking on 2 feet first, because this would very much be indicative of a bipedality first stance. That's why she noted that they were so human-like, because it was a big find.
Right. So I do understand and agree with what you're saying, that there were disagreements. I was under the impression, I believe, that she definitely didn't at least, designate it to Donald Johanson's Australopithecus afarensis. And I know that there were some disputes whether or not all those bones, the footprints, did they belong to one species or was it multiple species? But at the end of the day, if the evolutionist does have some type of explanation regarding the modern human footprints found in Laetoli or found in Crete, I just find it funny because evolutionists are always asking you, where's the evidence that Homo existed in these layers with our so-called ancestors? And if we point them to, say, footprints or some bones that are indistinguishable from, say, the Homo genus, then usually there's a circular argument that has to explain it away instead of just admitting, okay, there is something that looks like Homo existed during this time. So it just seems like there's a circular argument because the evolutionists can't admit that, yeah, these are human-looking footprints because they don't believe that humans lived during this time. So it just seems like a circular argument. I'm not sure if it's something that just needs to be agreed to disagree upon regarding the footprints, but I definitely like to see the paper that you talked about regarding and if it was indeed not a footprint, because I haven't seen that, and then also the information on Leakey and what she concluded.
Yeah. Let me send you after this conversation. I will send you. I've got some awesome papers just like on the back burner that I can kind of just send to you so you can look through them yourself. But
Awesome.
It's not so much. You mentioned that it's like show me the evidence, and then we find what a potential footprint in Crete, and then human-like footprints in Laetoli. Well, no one ever thought that those were human footprints. I would challenge you to find anything by Mary Leakey saying, yes, they are certainly human footprints, or I think that they're human. Not, wow, they look really human, because they look very human, aligned with one of the primary hypotheses at the time, and it turned out to be the prevailing one. That's what we abide by today. When we look at the hominids, we see all the traits for bipedality show up way before the brain cases start exploding in size. So I don't think that it's people ignoring the evidence. This is just the natural progression of things. The Crete thing, yeah, I can send you what I got on that for sure. Because I gotta say, if your evidence that there are hominids living in Europe, and that one of them made the Crete footprint, if your evidence for that is something that may be a footprint and may not be, is all you got, considering how well things fossilize all the time elsewhere, which isn't to say they fossilize very easily. I'm just saying, to find no trace of any hominid there, rather any Australopithecine and back hominid, I don't find that particularly convincing.
Right. It just seems based on findings all the time there is. I mean, even now they agree that there's more of a bush instead of a tree regarding the, ape to human evolutionary ancestry. And then regarding certain things like fossil footprints or Crete, from what I read in the primary, source data, they are concluding that they are footprints, but, who actually made them. So I think it just comes down to the fact that I'll have to look at, the paper, the material that you're suggesting. That maybe they're not human footprints, but I think even moving away from that topic though, because I am really genuinely curious if there is any validity to human evolution. I think it's reasonable that, especially if you're in the affirmative, I've always struggled with the evolutionary out of Africa theory, because it sounds like it really does involve an extended near extinction event, but that extinction event would be associated with severe inbreeding. So for one, do you hold to the out of Africa scenario or the multi-regional scenario, and kind of go from there and maybe give an explanation as to how that might be feasible regarding Homo erectus and then Homo sapiens?
Well, generally speaking, at least as far as I know, the current consensus on things, because as far as what I think, I try to go off of my impressions of the current literature. And the literature changes, because you're right, that is the nature of science. Like the time scale has been swapped around several times in light of new information, which is kind of how science should work, at least in my opinion. You always want to be closest to the truth. But things have been changing less and less all the time. Now it's slight tweaks here and there. And as far as I know,
Right.
the general trend of the gradient of hominids has gotten pretty solid. I don't think there's been a large change in that in quite some time, other than the finding Homo sapiens quite a bit older. I think it went from 200,000 to 300,000, which is very neat. But I digress. As far as the out of Africa theory and multi-regional, I think what's currently going on, and that's not my specialty. In fact, I found that part slightly less interesting than the hominids and the morphology itself when I was taking human evolution. But I think the current consensus is that there were, I think, 3 kind of bursts where you had erectus leave, right, and diversify into floresiensis and denisovans somewhat far away.
Yeah, right. I believe even and sorry to interrupt, I believe in the
No,
out of
it's
Africa
okay.
scenario, there was 2 out of Africas. So maybe a couple million years ago, after Homo erectus evolved, Homo erectus left the continent of Africa, and then independently on the other continents evolved into, for example, exactly what you just said, right? The hobbits.
Right. Are you certain though
Denisovans.
that it wasn't Heidelbergensis that diversified into Neanderthalensis outside of Africa, into Neanderthalensis? Because at least what I am aware of is that we share a common ancestor with Neanderthalensis in Heidelbergensis, not Erectus.
Well,
So
right,
then
you
it
got
would
Homo heidelbergensis, Homo
Right. So then it would
neanderthalensis.
stand that there would be 3, right? There would be one where Erectus leaves and you get Floresiensis and Denisovans, and then there would be one when Heidelbergensis leaves and you get Neanderthalensis, and then Sapiens leaves as Sapiens from
Right.
Heidelbergensis, but staying in the
Now,
African continent.
so as all these hominins are evolving, right? So Heidelbergensis, Homo neanderthalensis, the hobbits, Naledi, obviously the Denisovans, for example, and then you've got Homo sapiens that are in Africa that evolve out of Homo erectus. They, according to what I've read in the primary literature, they would have had their migration, but that was also after the fact that they had a near extinction event, because we know that every single human being on this planet is no less than 99.9% similar, right? We have low genetic diversity.
Totally.
From my understanding, I'm almost done. From my understanding, the evolutionists were kind of caught off guard regarding that. So that's why they resorted to a population bottleneck roughly 70,000 years ago that would have reduced homogeneity, right? But my question to you is, how could such a tiny, nearly extinct, obviously, Erica, they'd be genetically compromised due to inbreeding, prolonged inbreeding, suddenly explode into all parts of the planet like you talked about, and then seizing dominion over the world, whether they assimilated with all these other hominins, you know, Heidelbergensis, hobbits, Naledi, for example, which obviously hobbits and Naledi, I conclude are just humans that are genetically compromised due to inbreeding and isolation post-Babel. But if there's any validity or feasibility to human evolution, how do you explain those humans diverging from Africa, even though they should be so genetically compromised they should have gone extinct? Go ahead.
Well, no, not necessarily at all, actually, because the bottleneck that humans are thought to have experienced due to the Tobu eruption.
Right.
Hold on.
Yeah,
just to be sure that. Okay. Go ahead.
Yeah, so the humans experienced, right, a bottleneck due to the Tobu eruption. Now, do we know of any other organisms that have experienced an extreme population bottleneck? Yes, we do. Everyone loves cheetahs.
Right,
cheetahs are essentially genetic. All of them are very close to being genetic twins to one another, which is indeed the 2 of them are much closer to one another, take any 2 random individuals than any 2 random humans. So I would say, yeah, it looks like they did an A-OK job recovering from an extreme bottleneck that was much worse than that of humans. My question to you would be, in response to that, because my point being, I don't think it's any problem having a population bottleneck. I'm fairly certain that the population wasn't even that. It was pretty small, but it wasn't that small.
How small would you say? I'm sorry to interrupt.
Before we go out on that rabbit trail, just to be sure that that last point is wrapped up.
Could she just say, is it 2,000 or is it 10,000? I've heard some evolutionists say 2,000 people, other evolutionists say 10,000. I mean, what do you hold to?
I don't know. I don't know the literature on that. I know about the Toba extinction, but that's not, like I said, out of Africa and that particular bottleneck aren't my specialty. I could talk about morphology all day.
Hold on, hold on, Stanford, just to be sure that with that before we move on to in case you didn't finish that last point, Erica.
Thank you, James. I was just going to say I don't know for certain, and that my kind of reverse question to you would be, if you're pointing to the bottleneck as being a result of kind of the flood, my question would be, all right, why don't all species have such a severe bottleneck? And why do the cheetahs also have a bottleneck? Why is it humans and cheetahs? What's the model for that?
Okay, very good questions. I'll answer those questions. I want to kind of object to what you said. I find it funny every time I ask this question to evolutionists, I do find that they fail in answering it, because they always bring up the 10,000 cheetahs that we see in the world today. We know that conservationists, do feel the cheetah is already showing serious signs of inbreeding and genetic decline. I mean, there's just not enough of them. And it's a fact that genetic diversity in these cheetahs has eroded. And what they're seeing now is the expression of many destructive mutations. So all the answers that I get and that I find in the primary literature regarding this out of Africa are not credible, because small bottleneck populations have extreme problems. And the thing is, this bottleneck would have been prolonged, due to the inbreeding, due to the genetic compromised population of 2,000 or 10,000, they would not have been able to seize dominion over the world. That's a fact.
So why though?
Okay, unless you want me to answer. So was your question, how do we?
Oh, sorry. Right. Go ahead. Yeah. Go ahead. You can. Yeah, but go ahead. That's fine.
Okay, so why is there not? Actually, if you can rephrase your question. So is there a problem with our bottleneck after the flood? Is what you're saying, like due to inbreeding or?
Well, I'm saying if you've got if you have a single pair of multiple individuals, you should have every single organism that resulted from those kinds essentially looking like the cheetah right now. Much worse, actually, if you're going from a single breeding pair. I mean, we consider that functionally extinct in ecology today. I mean, it happened with the northern white rhino. That is an unrecoverable loss of genetic diversity. Because you're right, genetic diversity absolutely matters. The cheetahs are experiencing trouble, but I don't know that I would phrase it as that they're experiencing, a higher rate of mutation. The problem is that their lack, I mean, maybe I don't know enough about genetics to say, but as far as I know, I thought the primary problem was that they're all so identical that if there's a single disease that hits them, they have no defense because there's no kind of variety, which is really important. But yeah, that was my question.
Yeah, and I think it all comes down to the fact of where does genetic diversity come from? Because the big difference between my view and then say your view, Erica, was that I explained the vast majority of DNA differences, right? For example, nuclear DNA differences, not by mutation. And that's what evolutionists explain them by, right? All the millions and millions of DNA differences they say are the result of mutation. But according to our model, we explain them by pre-existing or created DNA diversity. And that one genetic difference between your model and my model has massive implications for the timing as to the origin of species, for example. So if we have these differences from the start and therefore no mutation accumulation by the time we reach the flood and the population bottlenecks, for one, very little heterozygosity regarding the original created heterozygosity wouldn't have been lost. There wouldn't be any bottleneck problem. We wouldn't expect to see the same thing that we see with the cheetahs. But regarding your model, you guys do explain the vast majority, if not all, the DNA differences as the result of mutation over time. So mutation accumulation and the results of inbreeding pretty well makes your out of Africa scenario impossible. And
well, I don't see why though.
Reasoning, but nothing really that's feasible or valid. So that I'm destroys the model. Go ahead. Take it.
Sure. Sorry. Yeah. I was just going to say, I struggle to see why you're having a problem with that. I mean, cheetahs aren't doing the best that they could, but they are certainly not doomed to extinction, and they have a much tighter population bottleneck than that the ancient humans are proposed to have had. So I don't understand why you're saying, I mean, the cheetahs did it and they're still doing it, and the humans' problem was less. So I don't see kind of like, I feel like you're kind of saying, oh, it's a problem, but then it's like, why is it a problem? You're not providing a reason other than saying, well, it's bad, there'd be inbreeding. Well, there might have been, but it clearly wasn't too much of an issue. I mean, that's kind of like
exactly why you say it must not have been an issue, because you have to hold to that primary assumption that ape to man evolution actually occurred. But the reason why is because that evolutionary bottleneck hypothesis, like I said, it involves an extended, that's the problem. It's extended. It's not one generation like our bottleneck. It also involves a lengthy near extinction event. So that is a fairy tale, because that hypothesis is associated with severe inbreeding is not even remotely feasible. What you said about the cheetahs, yeah, conservationists are trying to keep them alive due to the recessive mutations that are being fixated in the populations. I mean, their sperm is degenerate. They probably will go extinct. They would have gone extinct if it wasn't for conservationists. But this,
not at all.
2 to 10,000, well, this 2 to 10,000 population of Homo sapiens that came out of Homo erectus would not have been able to seize dominion over the entire planet. Plus, there's evidence that all of these populations you're talking about, right? I liked your analogy on your last debate about Middle Earth, because I love Lord of the Rings. Yeah, it was kind of like that. You got Neanderthals, hobbits, Naledi, Heidelbergensis. You've got the Denisovans. You've got them all living at the exact same time.
So I guess we can just kind of leave it at agree to disagree. I don't particularly see a problem being, with such a bottleneck, considering that we've seen others. I would like to, if it's okay, since I would like to maybe take a turn picking a topic, I would like to talk about chromosome 2, because you brought that up in kind of a, it's been overturned. I would say not in the slightest. Let me fix this here. My thing fell.
I just want to ask the moderator, because, obviously I want to talk about chromosome 2 fusion. I know we had a 30-minute discussion. I want to know how much time we do have to talk about chromosome 2. James?
I've got flexibility with time if you guys do, but that's...
Yeah, I could go all night. I'm good.
So I think we've got enough time to go, we go for another 45 minutes, including Q&A, which would be 25 minutes at the end, maybe even 30. So I would say if we go maybe for discussion-wise, maybe another 15 minutes, and then go into the Q&A.
Sure.
Sounds
Perfect.
good. Yep. Okay. So
Okay,
what would be?
so-
Yep. Okay. Go ahead with the chromosome 2 fusion. What would be your objection, I guess, to the claim that I made overturning it?
Okay, so, this would be kind of it, because Jeffrey Tompkins is known for being the guy when it comes to debating the human similarities with chimpanzees, which I did look into that 88% claim, and it is somewhat, I hate to be kind of sassy, but it's very abysmal. First of all, he used an extremely buggy version of BLAST when he got that first 70% number, and no one can seem to get that 88-ish% number when they're running BLAST how it's run in every single other species. It was kind of, as far as I know, it was revealed that he was essentially saying, all right, if you have a strand of DNA and there's a single base pair difference in the middle, that goes from being 100% identical to being 50% identical, which is very interesting that he's choosing to run it that way with humans, considering we don't run it that way with any other species. But with regard to human chromosome 2, essentially Tompkins argues mainly that it's not a fusion site because there is a functional pseudogene at that site. Now, first of all, it would be functional in that it is indeed expressed. It's barely expressed, first of all. I mean, it's barely interpretable from noise in the background. But yes, it is there. But there's also a promoter.
Okay, wait a minute before you go on too long, because I just want to touch on that part that you just said.
Sure.
Because you're right. We do look to that reputed fusion site, and we're saying that it's overlapped by a functional gene, okay?
Yeah.
So that so-called fusion site is, we're arguing that it's not actually a fusion site at all. It's indeed a functional component of that DDX11L2 gene. But are you aware that so-called fusion site does lie within the first intron of that gene, and that it also is an active promoter for the transcription of an alternative transcript, Erica? It also binds at least 12 transcription factors, and it initiates RNA transcription, which begins with its own sequence, and it has a chromatin profile that further supports its active promoter status. So why do you say that there's barely any function and barely any activity there, when these are the facts that we understand?
Well, you say all those things as if they're this grand and vastly vital process. But if that were indeed the case, and I'm not a geneticist, so I don't know this for certain, I would imagine that it would show up as more than just noise in the background. I mean, you had to scrutinize that this portion of the genome to essentially even find that there was a pseudogene there. And furthermore, are you aware, Standing, that there are the calling card for telomeres right at that fusion site, constantly and consistently repeated, this both the forward, which is TTAGGG, and of course the reverse, which is CCCTAA, and they're flip-flopped just like they would be on the two opposite ends of a telomere. So I don't, and as for there being a functional gene, it's also been.
Hold on one second. Let's just.
It's just because genetics.
Hold on, okay. I know. Just to be sure that we get plenty of time, we'll let her wrap up. If you can wrap up this point, Erica, and then we'll kick it back.
Yeah. Sorry. Yeah, I was just going to say, furthermore, there just being a pseudogene there that is indeed functional. We see functional pseudogenes on fusion sites in other animals that have been accepted to have had telomere chromosomal fusion, even by creationists. This is a paper that was done on equids, where it does indeed happen. So I don't, and I can provide that paper for you too if you'd like. But essentially, as far as the conventional science goes, there is like actual empirical evidence that promoters can cause pseudogenes to essentially relocate onto a fusion site. So I don't understand, essentially everything rests on this being a functional gene, but that's not problematic. And it's been shown in other animals to be A-okay.
Well, I don't think it's just because of that, because you did talk about the telomeres, right? The ends of chromosomes. And
Yes.
I'm sure you would agree that telomeres, they are served in our genome as end caps that actually protect the DNA. And one of their jobs in protection is to prevent fusion to begin with. So why were they even involved in this so-called reputed fusion if one of their jobs is to actually prevent it? And from my understanding, this apparent hypothetical fusion, which obviously we can't observe happening, two questions. Why can't we reenact it in a lab today? We don't have any DNA from fossils to see how it happened, if it actually happened. And lastly, it's not really evidence for ape to man ancestry anyways, because even if I conceded that there was a chromosomal 2 fusion, as you know, we've got the chimp and human ancestor who split, and then in the human line is when this supposed chromosomal 2 fusion occurred. So either way, it's indirect evidence for ape to human evolution. So that's no help to you. And secondly, we have all these discrepancies showing that possibly it isn't even a chromosomal 2 fusion site to begin with. So it's kind of like 0 for 2 for the evolution.
Well, but the problem for that is there aren't refutations. Like I said, this is a known phenomenon. I mean, this paper was in 2013. We've known this for 6 years, that this fusion sites can indeed hold functional pseudogenes on top of them too. Why it's important is because creationists for the longest time were like, well, there's a chromosome difference. They can't be related. Well, yeah, they can, because what if the chromosomes fused? And look, they did. And this was a prediction that was made and met. And the only as far as what you've said here and what I've seen from Tompkins, the only essential refute isn't even a refute. It's basically a guy saying, well, I have a problem because sometimes pseudogenes are functional and there is indeed one on top of the fusion site. But then here it is, not problematic in equids. And oh, sorry, go ahead.
Is this specific telomere to telomere fusion, is this of its kind? I know there's been other chromosomal fusions, but this specific one that they're purporting, is that the first documented type ever seen in nature?
I wouldn't imagine so, since it happened in the equids.
Right, but the one in the equids, I believe, and I don't have it with me, the paper, I believe it was a totally different chromosomal fusion than the one that's being purported here, right? This telomere to telomere fusion. You didn't really answer my question regarding telomeres anyways, how they're used to protect the DNA, and one of their main jobs is to prevent fusion. So why should we believe that they were involved in a fusion? But this is a hypothetical fusion too.
Sure.
We see these chromosomal fusions happen in nature that I believe aren't the same as this reputed one. But why are we to believe, just like the out of Africa scenario, which I don't believe you answered as well, which kind of destroys human evolution, why are we to believe that actually occurred? Because it's all theoretical, hypothetical.
The human too? Well, here, I'll let you pick. Would you rather me answer out of Africa, which I thought I did, but maybe I didn't. I might not have, genuinely, I can't remember. Or would you rather continue with human chromosome 2? You picked the path.
Yeah, you did answer the out of Africa, but kind of like you accused Hovind of answering your question, but then you said that, he didn't really answer it sufficiently. That's what I'm saying about the out of Africa, because there really is no feasible way for this 10,000 Homo sapiens to seize dominion over the entire world after a prolonged bottleneck of severe inbreeding, the accumulation of mutations. I mean, it's just not feasible. It's fairy tale. When it comes to the chromosomal 2 fusion, I just want you to answer the question. If telomeres, one of their jobs as being the end cap of chromosomes, is to prevent fusion, why are us to believe that somewhere down the line, millions of years ago, after the split between chimps and humans, this purported fusion actually occurred, and that it's unlike anything we've actually seen in nature regarding fusions? I mean,
okay, hold on. Just so we got to wrap up this point, saying for truth, just so we can kind of keep it going back and forth.
Yeah, sure. Standing, you want to finish that, or you want me to go ahead?
No. If you just want to, I guess, answer that question regarding the telomeres.
Yeah. So with telomeres, why are there mutations at all? I mean, our DNA, when it's actually replicating, there are checks and balances that are supposed to prevent mutations from happening that fail all the time. That is the nature of genetics. So yeah, telomeres are supposed to protect from that, but it obviously doesn't do it all the time. Now, as far as this not being the same kind of telomeric fusion, I'm not sure. I'm pretty sure it is when I kind of eyeballed the abstract, but I can't say for certain. Genetics is not my forefront. But I would like to talk about out of Africa, because I do know enough about genetics to say that we have a real-time example. I keep bringing up the cheetahs because I'm not understanding like why it's problematic for humans. But
I don't want to interrupt, Erica.
Hold on.
I want to go back to the
one sec. Just to be sure.
Okay.
Hold on one sec. Standing for Julie. Just to be sure that because want to be sure that she's got plenty of time to respond, because I think there were two questions.
I'm just trying to answer.
Oh, right. You're right. You know what? I apologize. I did ask the out of Africa one again. So
yeah. I'm just trying to get them both in.
Chromosome 2.
Yeah. I'll quickly just rehash out of Africa, and then that's kind of why I was like, let's pick one, but we can try to juggle both. That's fine with me. With out of Africa, right? I mean, so I'm going to really just like make my point as kind of concise as I can. Humans have a bottleneck. Cheetahs have a bottleneck. The cheetah bottleneck was much worse than the human one. And you don't have to be there to know that, because all cheetahs are more closely related to one another than all humans are today. So know that they're less genetically diverse because of a bottleneck. Okay, great. Cheetahs did very well for themselves until humans started hunting them and destroying their habitat, which are the main reasons why there are less of them today than there used to be, not because of these new genetic mutations popping up. As far as I know, anyways, I'm not a cheetah geneticist, although that would be a cool job. With humans, it shouldn't be any more of a problem. I mean, essentially, cheetahs had a worse situation and did just fine until something completely unrelated hit their numbers hard. So with the humans, and also, mind you, the Toba extinction or the Toba eruption was indeed a volcanic eruption in nature. So while, yeah, I think that the estimates are around 10,000 individuals, that isn't certainly the case that there weren't other bands of humans out and about. Now, granted, it probably wouldn't change that number very much, because as you know, genetically, that number is genetically kind of constrained for a reason. But you're essentially saying it's impossible, at least this is what I'm gathering. You're essentially saying what my ears are picking up, that humans had a bad bottleneck, thus they shouldn't have been able to spread out and essentially diversify across the planet. Except cheetahs had a worse one and they diversified across Africa. So that's my answer to out of Africa. I don't see it as an issue. I don't think really anybody does. I've actually never heard that argument before. And I look at a lot of creationist arguments. Anyways, that's my answers. Those are my answers.
Awesome. Okay, if I could just say a few things. So the cheetahs, for example, are being looked at from conservationists today. Inbreeding is the issue. We know inbreeding exposes the genetic mistakes within the genome. And I always say regarding genetic entropy, inbreeding is like a sneak preview of where we are going genetically as a species. I mean, we accumulate 100 new mutations per person per generation, most of them being nearly neutral. What we now know about the function of the genome, that means we're degenerating faster than we ever thought. Now, this population bottleneck in the out of Africa scenario was conjured up by evolutionists once they realized that human diversity is very, very low, indicating that we must have come from a very small population. That small population, according to my belief, obviously is Adam and Eve. That makes sense. God created Adam and Eve. What do we see today? Low genetic diversity. Me and some man over in Asia, no more than 99 or no less than 99.9% similar. That makes sense. Evolutionists has always had to come up with a rescue device. Yeah, there must have been some kind of genetic bottleneck 2,000 to 10,000 Homo sapiens, say 70,000 years ago. The thing is, this was a prolonged bottleneck involving severe genetic damage due to inbreeding. Because when you get small populations like that breeding together, not for one generation, not for 10 generations, but many generations, as the out of Africa scenario would suggest, that population would have been so genetically compromised that there's no feasible way that they could have seized dominion over the entire world. So I'll let you comment on that if you want. But the chromosome 2 fusion, I just want to mention with the chromosome 2 fusion, You were talking about the telomeres, and we should see remnant telomeres. But from my understanding, there's, for one, a struggle to find these. That area is very, very degenerate. Why aren't there any more sequences? I think it's not just the fact that the purported fusion site is actually a functional DNA element in a human gene. There's other discrepancies that go along with it. So not only that, not only the telomeres, but also the fact that remnant centromeres are also difficult to find. So it's not just the fact that a gene has been found and it spans the fusion site. It's a number of things. But like I said, it's still indirect evidence because this supposed chromosomal 2 fusion occurred in the human line, which means we could easily explain it based on either pre-flood or post-flood, especially because we believe in a population bottleneck anyways. It would have been passed on to everybody we see on the planet. So it's just not very good, convincing evidence anyways for ape to man evolution. I just think you're going to need something better. The pseudogenes you talk about, we know that they have very important functional roles in immune response. They're necessary to sustain healthy life processes. So go ahead.
Yeah, sure.
Actually, take as much time as you want, Eric. I talked a lot there. And
yeah. Well, I'll introduce something quite interesting in a second that you've heard before. But one, the reason we do find remnant telomeres, this is actually Tompkins's research here. So those are the remnant telomeres that you're looking for in that central part of the chromosome. With regard to, kind of pushing an extinction event, we know the Toba extinction event happened, I'm fairly certain, because there are ash beds. There are enormous ash beds. And that's how we know that it was a really bad extinction event, or really bad eruption, sorry. And really bad eruptions tend to not be super good for the things that are indeed around them. We know that from all the volcanic eruptions that we see today. That would be my sort of answers to that, but to kind of take a swerve, and feel free to answer those if you want. I mean, I think we're just chasing our tail with that. We're just going to disagree. But I'll ask you the same, you brought up my question to Kent, so I'll ask you kind of the same question. If we use genetics, right, to determine relatedness within the human population, why can't we do it between populations? Because I find that quite compelling. You don't find that as genetic evidence very compelling. I was mostly using it as a refutation because creationists often say, oh, well, there's a chromosomal difference. Okay, well, it's a fusion. But the fact remains that that's not really the point. And it's also not just the fact that there's a fusion, but that the tops and bottoms of that chromosome are very, very, very similar to 12 and 13 in genus Pan. So where do you draw the line? I asked Kent. I didn't really get an answer from him. So where do you draw the line?
So where do we draw the line on common ancestry?
No, where do you draw the line between humans and chimps? Why is it okay to use me and my mother's genome to confer a common ancestry between the two of us, but not between me and a chimp?
Right. So if we're looking at, for one, we'd have to look at DNA differences. It also comes down to the fact that humans, okay, for all of recorded history, have produced humans. And we can use mitochondrial DNA, or we can use the Y chromosome DNA, and we can test whether or not, or we're related to our father. We can actually prove that the entire, all of humans, the entire human species are related. This is verified through independent corroborating evidence. Okay, but now you want to extrapolate that to pretty well suggest that humans and chimps are also related, even though there's so many discrepancies. Because I always say there's no point in arguing over evidence that's kind of agnostic to the debate. And what I mean by that is genetic similarities, whether it's nested hierarchies in DNA on a molecular level, mitochondrial DNA, for example, nuclear DNA, or nested hierarchies regarding anatomy, morphology, physiology. This can either be explained by common design or common ancestor. We need to look for evidence that can differentiate between the two. And I believe there's plenty of lines of evidence that can differentiate between the two. For example, the Y, just one sec,
yeah,
the Y chromosome. Okay, so this is going to be my follow-up question to what you said. Let's look at the Y chromosome, okay? Evolutionists were shocked. The Y chromosome is the most stable chromosome in the human genome because it doesn't exchange its genetic content with anything else. It doesn't experience recombination. Yes, gene conversion, of course, amongst itself, but this should be the most similar chromosome in the entire genome regarding apes and humans. Why is it less than 70% similar?
Am I good to go?
Yes, take your time.
Okay. So I feel like that's a little bit of as Kent would put it, like a hit and run, right? A slap and run, because that didn't really answer the question. I mean, you basically said humans produce humans, therefore it's reasonable that genomic comparison between me and my mom would be common ancestry. Okay, well, what if you had an individual, an orphan? You don't know where they came from. You didn't witness their birth, but they certainly have a similar genome with the mailman. You didn't see it. You don't know for a fact that that is indeed the father or the mother of this child, but their genome is very similar. So similar, in fact, that it mimics that of mother and offspring or father and offspring in other groups. So I think you would indeed point to that and say, okay, well, common ancestry. And that's what creationists do within their kind groups when they're formulating their kind groups. My argument with this isn't necessarily to say, whatever, pine trees and elephants are related, but to point out that there's no way of getting around lumping humans with the apes, because we use in a court of law genetics as a means of relatedness. I mean, you're basically saying we should draw an arbitrary line because we have never seen a human produce a non-human, which even if they did, that would violate the law of monophily. But yeah, because recorded history has been around for 10,000 years, less. And even then, it's barely been recorded. I wouldn't suspect no one under an evolutionary lens would suspect that you would. So I feel like that's somewhat of a non-answer. You showed the transitional designs earlier. Okay, well, where's the plane with wings that can't let it fly, or with half of an engine that performs a different function? I mean,
okay, here, let me respond to that real quick. I think you went.
Yeah, sorry, I did dodge around. around.
Yeah, no problem. So here's my answer. That's not quite as technical as what I said regarding genomics and genetic data that
Sure.
can differentiate between the two. For one, as I've stated, we would expect nested hierarchies, whether it's anatomical, morphological, physiological, or genetic related hierarchies. But regarding your question of where do we draw the line, why can't we conclude that apes and chimps are related, but we can conclude that me and my grandmother is related? Well, the thing is, we need independent corroborating evidence. Okay, so not something that also doesn't rely heavily on assumptions and inference. That's what I'm saying. There's an extrapolation fallacy there. So here's one example before you go. We have DNA testing, like you're talking about, that confirms, okay? There's no argument. Nobody argues, I hope not, that all humans are related, okay? Also mother, father, grandmother, we can see this based on maternity tests, paternity tests, but these various testimonies are completely distinct from DNA, okay? There's also corroborating, because we validated this test over and over and over again in humans. Like you've said, all of recorded history, humans have produced humans. But unfortunately for the evolutionists, okay, now you want to extrapolate that to mean humans and chimps are related. Well, we can't go ask the chimpanzee, we can't go ask animals from millions of years ago, or even animals from today, for their testimony regarding the DNA testing of them and their offspring like we do with humans. So there's just no way for us, Erica, to have independent corroborating evidence here regarding your question. But we do have independent corroborating evidence when it comes to all humans being related. You're committing an extrapolation fallacy.
Just to keep it short and pithy, standing for truth.
Yeah, 100% agree. We do know that all humans are related. Why do we know that? Because our genetics are so similar. So, why do we know that humans and Neanderthals are related? Well, because their genetics are so similar. You can take that back down the line, but creationists can't come up with anything empirical to draw the line. You say, well, we don't see it. We can't ask a chimpanzee. Okay, well, we can't see the inside, or rather how a black hole functions in real time, but we can make accurate predictions about how light's going to react around it because of the data that we do have. So there is no reason why you can't extrapolate it backwards, any more than there isn't a reason why 23andMe can take your DNA and take your DNA and extrapolate backwards and say, guess what? You're 63% Norse. Well, why couldn't they? I mean, you're essentially kind of saying that this is impossible because no one was there. It's an appeal to observational science, which I'm not a very big fan of. And then you say, where's the corroborating evidence? Well, it's all the other stuff I showed. It's the fact that humans are morphologically nearly identical in so many ways to all of the other apes. It's the fact that we have this smooth gradient, and creationists can't come up with a meaningful way, at least in my opinion, to say that we can have this accelerated decay that completely throws 4.8 billion years out the window. It's the archaeology, the fact that no one can explain why there's so many dang tools all across the surface of Africa. All of that points in one direction, and it isn't to 6,000 years. So it's kind of like parsimony, right? What's the most parsimonious explanation? Because I'm not going to sit here and tell you that we know for sure exactly how things went, because we don't. And I don't think we ever will. I would love that. That would be awesome.
Okay, can I respond to a few things that you said?
Yeah, sorry, I'll wax poetic. I'm guilty of the same thing.
No, I think we might be both guilty regarding that. So I'm not saying that it's only an extrapolation fallacy that we can look at similarities in our genetics. Like I said earlier, every single human being on this planet is 99.9% similar, which says we have low genetic diversity, indicating we came from a small population. Okay, this is a good thing for us. Now, you want to extrapolate that to mean we also have similarity with chimpanzees, for example, and then we also have similarities with monkeys, we have similarities with dogs, whales, fish, all the way down the line to a bacteria, the nested hierarchy of life. I'm saying we too predict nested hierarchies. Therefore, it's difficult to draw that line since we both predict the exact same thing. We need something that's going to differentiate between the two lines of evidence that both models can explain. That's why I pointed to something, because you talk about similarities. The Y chromosome, for example, and this time hopefully you can answer it. But the thing is, the low level of recombination and sequence diversity on the Y chromosome that I'm talking about should make the Y chromosome the most similar between humans and chimps. But the problem is that the human and chimp Y chromosomes are actually incredibly dissimilar when they should be the most stable, because they are immune for the most part to recombination. So how do you explain the fact that the Y chromosome is less than 70% similar between chimps and humans?
Well, first of all, I would, as I've continued to kind of begrudgingly admit throughout the course of this conversation, that genetics really isn't kind of my field. I can talk to you a lot about other stuff, but genetics isn't it. So I'm not kind of adept enough to really answer your question in a way that I feel that you would find sufficient. I don't know. I would ask you out of curiosity, if by chance that 70% number comes from Tompkins, considering Tompkins.
Nope. I've had a good question. I have the paper where it came from.
Do you know who the author is?
Source data, because they were shocked by it. But from my understanding, talking to scientists, where I've debated Dr. Stefan Frello, for example, their explanation would be gene conversion, because it can exchange genetic material within itself, right? It's like recombinations, like shuffling a deck of cards, but the Y chromosome doesn't have a counterpart to do that too. Therefore, they should be the most stable. It's the best chromosome to look at. My question would be to the evolutionists like Dr. Stefan Frello or whoever, Herman Mays, that wants to look to that as a rescue device. What testable prediction can you make and bring forth regarding change on gene conversion in the Y chromosome? Because and it's okay that you don't have the answer to it, but that would be a differentiating line of evidence that can tell us, are chimps and humans actually related? There's no way they can be, based on the dissimilarity of the Y chromosome. Less than 70%. Evolutionists were shocked, Erica. Shocked.
But here's the problem with that. That's kind of essentially hinging it all on the fact that I don't have an answer for the Y chromosome. Now, what I do know about genetics would be, even if the Y chromosome were 70% dissimilar, well, there are some proteins between humans and chimpanzees that are like 50% similar. And the reason that they're 50% similar is, as we know the mechanisms of genetics can kind of swap things around. But the fact remains that we are still the closest related to chimps with regard to that single protein, even if you're just comparing that single protein across, the animal kingdom where that protein has been kind of examined, would still be the closest to the chimpanzee. It might be only 50%, but you're not going to get any closer. You're not going to find another animal. If you could find a rabbit with a greater similarity, well, then you might have something to kind of go on, unless it was, say, like a remnant that had been turned. If you'd been able to kind of show that it was a de novo type situation, but the fact would remain I bet, and I don't know this, but I would put a small amount of money on it because I'm not very wealthy, that the Y chromosome, even though it's 70% similar only, that's still the closest member of the animal kingdom to humans. And it may simply be, as you know, there are areas on the genome that are kind of hotspots for mutation.
Mutational hotspots, yeah.
Yeah, sure, yeah, and they just kind of undergo it quicker for whatever reason. I'm not a geneticist. I wish I could tell you. I wish I knew the answer. But my answer would be, how do we know that something similar is not going on the Y chromosome? Can you show me an animal that's closer than that would kind of violate it? Because as far as what I know about all of this stuff, that's still not enough to say humans aren't apes, aren't a member of the ape kind. Would you be the type of creationist that would say, no, humans aren't primates? Because I know Evolution News admits humans are primates, and I know Creation.com admits humans are primates. Even if they don't like common ancestry, they admit that morphologically and genetically, we should be classified as primates. AIG, they don't like it. But where do you stand on that? Where do you stand on that? Do you think humans are primates? And, if not, why not?
Okay, so first of all, with the proteins and Y chromosome comparisons, before I answer the question, just to address
sure.
Things.
I went on for a while.
No, and that's okay. The whole point of looking at the Y chromosome, say, versus the proteins anyways, is because the Y chromosome is known scientifically to be the most stable of chromosomes. So that would make sense to look at that to see, how close the similarities really are between humans and chimps. But I don't want to beat a dead horse regarding your question regarding ancestry anyways to begin with.
And then forgive me, sorry to interrupt. Not you saying for you didn't do anything wrong. I just forgot to mention
for once
we will go to Q&A pretty quick. So if you're both able,
don't we have a quick closing just to wrap things up?
Forgive me if I forgot that from the email. Was that an email?
I'm okay without a closing.
Oh, deal.
Even just a few minute closing just to wrap things up.
Sure, you betcha.
I guess to answer your question, so are we just going to kind of wrap this part up here and then jump into closing and then Q&A you were saying, Jay?
Well, I suppose what we could do is, if it's something you can fit into your closing, we go into closings. But if it's something that you're like, oh, this is just kind of really relevant to this point right now, we can do it.
Okay.
Just short.
Actually, that's good. Yeah, I can
50.
wrap it up in 30 seconds, and then if she wants to have the last word, then we can jump into the closing. So I think Erica, it all goes back to the question anyways, as to how we identify common ancestry. Okay, so if we both predict nested hierarchies, then we need to look at something that can differentiate between the two models. Now, we explain anyways the majority of DNA differences based on our model, which does lead to testable predictions that are future and novel as pre-existing functional DNA differences. Now, we only know very little of the DNA language. We speak less than 1% of the DNA language as far as I'm concerned. And all you got to do, Erica, is look at the history of genetics. I mean, in the 40s, there was one gene, one protein, one function. Science changes nearly every single day. I mean, we see massive amounts of literature giving us unexpected functions of RNA. And we've only functionally tested less than 1% of the genome because this gold standard of genome testing is gene knockout test anyway. So my technical answer, and to end it quick, I know we're going into concluding statements here, is genome function. Because evolution would say the majority of DNA differences, if not all of them, are the result of mutation. Therefore, we're looking at a lot of neutrality. We're looking at a lot of genomic fossils, right? Pseudogenes, for example, endogenous retroviruses, ALUs. Us, on the other hand, our model's based on pre-existing functional DNA differences. Therefore, we make a very specific prediction that the vast majority of DNA elements, DNA sequences, DNA differences are functional. This is a head-to-head prediction that we can test in the lab and see what is true, common ancestry or common design. That's my answer. Go ahead. You can have the last word, Erica.
Yeah, sure. I'm going to make my last word my closing. It's not so much does genome equate to function, it's genome does equate to relatedness, and there's no reason why that line can or should, rather, without empirical evidence, be drawn. I mean, essentially my whole point of this was that you have several lines of evidence that in and of themselves are convincing, but they stand alone. It could all be a coincidence, but together is when you reach a parsimonious conclusion. It's the combination of the fact that we can't accelerate decay. It's the combination, with in turn with the decay thing, in that we have this beautiful gradient of skulls that appear based off of the previous premise to have existed at different times. It's the morphology, it's the genetics, and it's, to be kind of sassy, the fact that no one can answer that question for me. No creationist seems to be able to answer the question without kind of reverting to, I know you like this word, rescue devices. So that's kind of all I got to say. I mean, I'd love to have another conversation with regard to whatever genetic entropy or the age of the Earth, because as I said to Kent, a lot of it hinges on the fact that I believe the Earth is very old and you believe the Earth is very young. Because obviously, if it's old, the skulls make sense, and if it's young, then it makes sense for your worldview. But it makes sense based on what we kind of think. So I wasn't convinced, Standing. You didn't convince me, unfortunately, and I'm sure I didn't convince you either. So, but that's okay. I still had fun.
Having fun is the most important part. You're right. You didn't convince me, but you're right. It was a good back and forth. We could probably do this all day. I appreciate your responses. I appreciate your arguments and objections to my position. I guess going into, so if that was your conclusion, James, I just want to set my timer here. How long do I have? Just a few minutes, or I don't want to go over?
A few minutes. I'd say roughly the same as what Erica did. So I think it's like, what, 2-3 minutes?
I'll be really quick because I just wrote. So in my opening, I tried to address nearly everything she talked about in her opening. I just got a few things hanging that I didn't get to address. I'm definitely a stickler for that. So I'm going to address the couple things here. So DNA analyses anyways confirm that the supposed Homo species now all coexisted, intermingled, and interbred, like we were discussing earlier. This is confirmation that they are all fully human. A few other things here that she discussed. Let me see here. Even at habilis, sediba, there's so much evidence now. Even some paleo experts are agreeing that these are classic examples of what happens when you piece together a so-called species based on a jumble of mixed bones. So what some evolutionists like Erica here are saying are your perfect transitional fossil from ape to man, it's actually a mixture of human and non-human bones. And they're simply an artificial species, a ghost taxa. That's why sediba, for example, Australopithecus sediba, is described as anatomical, with anatomical contradictions. For example, upper limb and shoulder anatomy, just like arboreal dwelling orangutans. And then you've got hands indistinguishable from modern humans. And this isn't the first time this has happened. This has all happened before. For example, one that comes to mind regarding the mixing of human and ape bones has occurred in Piltdown Man. So, which was just what they did. It was an ape jaw that was force fitted to a human skull. But unlike habilis and sediba, of course, I'm not saying habilis and sediba are a conspiracy by any means, but this was done intentionally, while habilis and sediba, not so much. For example, the evidence she brought in genetics, the similarity, I touched that. It's only 88%. Similar 400 million DNA differences exist between the two species. Pseudogenes have been overturned. Hierarchies, like we discussed, relative hierarchies, as I explained in the beginning, is just characteristics of designs. And then the Australopithecines, my last point. And then I think I covered everything. They share most of their features with living apes today. They're around 3 or 4 feet tall, with long arms and short legs, ape hands and feet, with long curved fingers and toes, opposable thumb-like toes, ape size and ape-shaped skulls, funnel-shaped rib cages with high-placed shoulders for suspensory behavior. And yes, they do have some uniqueness. I'll give Erica that in, say, morphology and anatomy, which is why the best conclusion is that these were apes and nothing more than just an extinct ape genus. And Erica probably knows the fact that a baboon bone was incorrectly assigned to Lucy for over 40 years. That doesn't really give me much confidence regarding the evolutionists and their many other conclusions on human origins. And I explained all the different species, Naledi, Neanderthal, Erectus, the hobbits, for example. These are all inbred subpopulations, explains all the degenerate features. Homo naledi, for example, explains the curved fingers. This is all expected after Babel, post-Flood, isolation and inbreeding. Everybody disperses. A few unfortunate subpopulations migrate, remain isolated. We see this clearly with the hobbits.
You got 5 seconds.
Dwarfism, for example. So I believe I've made a strong case that human evolution is impossible and fits perfectly within a biblical model. Thanks so much, James.
Thanks
you.
So
much, Erica.
Thank you. Time. So very excited, folks. We will go into the Q&A. So thanks so much for all of your questions. It's been a wild one, really enjoyable. I really like having both of our guests on. And with that, thanks so much for your patience, folks. I know you're eager, so I am as well. First up, Brian Stevens. Thanks so much for your Patreon question. They said, does Standing for Truth think that Kent Hovind actually has 3 PhDs? Also, where is his, Kent Hovind's, CV? Or basically, for those of you new to a CV, it's like a academic resume.
I'm more concerned with the arguments one has. Actually, Tija made that point once, and I respected it. You know, we're more interested in the arguments than, for one, ad hominems and looking at what people's so-called education may be, because some people with a lot of education may not have the best arguments, especially if they have nothing more than textbook answers. So I'm not really interested in responding directly to that. I'm just interested in the arguments that my opponent would have.
Gotcha. Thanks so much. Next up, we're going to try to speed through these folks. So if I'm going fast, forgive me. It's just because we want to squeeze as many in. Steven Steen, thanks for your super chat. He said, good to see Alpha Daddy, a.k.a. James, tonight. Gosh, you're sick. Nasty guy. You got to look out for him. Just, Next up, Michael Dresden, thanks for your super chat. They said, evolution is a myth, a fairy tale for grown-ups. A little trollish. Erica, if you want to respond, you can. You don't have to. It's up to you.
I won't give it the dignity.
Gotcha.
Okay.
Next up, Steven.
Good answer.
Michael Dresden. Oh, we got that one. Terry James, thanks for your super chat. They said, does different design mean different creator?
Does different design mean different creator? No, I mean, Honda, Volkswagen, for example, Chrysler, I mean, they have many different designs. You can look at the Volkswagen Jetta, which is a sedan. You can look at the Volkswagen Tiguan, or you can look at, for example, if you want to look at Chrysler, you can look at the Chrysler 300, the Chrysler minivan. I mean, there's different designs for the same creator. And of course, if God wanted to, for example, DNA, deoxyribonucleic acid is the universal code for all of life. And evolutionists say, this is proof that we're all related: pine trees, whales, banana plants, and apes, apparently. But of course, God is going to use a common code for his common design. If he used different codes, that would suggest a possible form of polytheism. Maybe there is multiple gods, like the Greeks or something. But one common genetic code makes the most sense given a biblical based God. So that's my answer.
Gotcha. Thanks so much. Jay Shy, thanks for your super chat. They said, Standing for Truth, genetic entropy is a lie by John Sanford. He misquoted Moto Kimura's their study where Kimura left out beneficial mutations on purpose to focus on neutral mutations by genetic drift, not selective mutations.
I feel like I've had this debate so many times. That argument is a rabbit trail. There's never been any rescue device or argument that has debunked genomic degeneration. All serious population geneticists admit and agree that man is presently degenerating. That's why they come up with rescue mechanisms, synergistic epistasis, mutation count mechanism. When I debate these evolutionists, they use the most weak standard blog related arguments. I mean, at least bring forth things like synergistic epistasis and these mechanisms that these population geneticists are invoking. Let's discuss the data. No more of these ad hominems. John Sanford shows a larger zone of no selection than Kimura because Kimura's analysis of the problem was oversimplified. I debated someone a few days ago who's very familiar in the very exceptional in genetics, and we discussed this very issue for an hour and a half. So no, these near neutral, low impact deleterious mutations, they are accumulating. But natural selection can only amplify the best beneficial mutations. It can only get rid of the worst detrimental mutations. Can't do anything against the near neutral mutations. They're accumulating in our genome, and beneficial mutations are no help. They are no help. The vast majority of them are unselectable as well. So there's my answer.
You got it. Thank you very much. And thanks so much for your super chat, Terry James.
Thank you.
They said, if DNA similarity can be explained away by similar appearance, then why is the, correct me if I get this wrong, thylacine more genetically similar to the numbat than the wolf?
Is that question for me?
And I feel like it is.
I think so.
It has to do with genetics and morphology. I
Yeah.
thought it was for him. Is it for him or is it for me?
I thought it started off for you. So now, if you can ask it again, I'll answer.
I think it's for standing for truth. I'll read it.
Ask it again.
Yes.
If DNA similarity can be explained away by similar appearance, then why is the thylacine more genetically similar to the numbat than the wolf?
I think, as I explained earlier, we're going to have to, if you're looking at just mere similarity and anatomy, morphology, for example, we need to look on a DNA level. And we speak so little of the DNA language. I am going to look to, like I explained to Erica, DNA function. And this has been proven true over and over again. Endogenous retroviruses, ALUs, pseudogenes, they're all very important in our genome. I mean, they're important to sustain healthy life processes in the cell. And I can go on and on about the numerous functions that have been discovered based on our hypothesis. But that example, I would if it's that similar genetically speaking, I would look to some form of function. I don't know if there's an answer yet to it, or if it's something that we need to discover in the future based on more genomic analyses and more genomic testing.
Gotcha. Thanks so much. Shannon Q says, Team Erica, in all caps. You've got a fan out there, Erica.
Shannon, I love ya.
Brian Stevens, thanks for your super chat. He said, Standing for Truth's sources for footprints is, quote unquote, the conversation.
No, I've got plenty of sources. Actually, too many sources on that one. Like I said, I looked heavily into that footprint. I'm looking forward to the source that Erica explained and is going to provide to me, because what I've found in the source data in the primary literature, they all agree that these footprints are modern, anatomically modern looking human footprints, and they're trying to explain it away with some form of possible extinct gorilla, or there's a lot of, I guess, data out there. So like I said, I'm looking forward to Erica's sources, but I've got a ton of sources. I can certainly provide them to anybody who is interested.
Gotcha. Thanks so much. Next up, Stephen Steen, thanks for your super chat. He said, my wisdom teeth fit in my jaw. Am I the missing link?
Hey, I had all four of my wisdom teeth too. Wisdom teeth are an interesting little facet because they're kind of on their way out, but not quite. Some people have no problem with them at all. I had problems with mine, and that was a wild after surgery experience. So I'll put it this way. You are the missing link. There's no other explanation. Gotcha.
Thanks so much. Next up, appreciate your question. Brian Stevens says, why do you ask Erica to use scientific... So this is for Standing for Truth. Why do you ask Erica to use scientific sources to disprove your blog from, quote-unquote, the conversation by a, quote-unquote, political analyst?
There's plenty of sources. I actually have innumerable quotes from all types of paleoanthropologists that agree with what I've been saying. I provided a paper in my opening regarding my hypothesis that these so-called human ancestors are nothing more than inbred subpopulations that experience rapid genetic entropy due to the accumulation of mutations. And we've got an evolutionist here who's written a paper recently pretty well confirming the exact same thing. Of course, he still holds to evolution, and he admits that there's something odd with the data, but I've got a ton of things that support me, quotes, sources. So if Brian Stevens is serious about it, I'll provide those to him after the debate.
Gotcha. Thanks so much. Next up, Terry James, thanks for your super chat. They said, Standing for Truth complains that Aaron interrupts, but it's okay for him to interrupt. How do you like them apples?
You know what? I think this was a good debate because Erica and I both, I think, like to talk a lot and talk about a lot of different subjects. But anytime there is interruption going on. James, being the best moderator on YouTube, stopped it right away. And thanks for that, James. So if R and Ra and I get in on this, hopefully we're not just talking over each other the whole time, because James is going to make sure that doesn't happen. So
that's
Is
nice.
that a planned debate? I'm interested.
Yes, it is. In fact, this is an excuse for me to show the thumbnail again, because I'm excited. This is going to be an epic one. So basically, RN Raw and Dr. Mick Leroy, I mispronounced it earlier, sorry about that. Dr. Mick Leroy and RN Raw will be debating on creation versus evolution, which best explains the evidence. And that's coming up in Dallas. It'll be in person. So we've actually... Team Skeptic.
That is so exciting.
It is. Team Skeptic, I have to give a big thanks. So if anybody out there, if you know Team Skeptic, give him a big thanks, because he was so kind. He's got a channel called Team Skeptic Debunks, or I should say, to be neutral. You can say debunks arguments without necessarily meaning that a view is true. He challenges the flat earth position. And he's a very good debater. And he's also very kind, because he helped us get the venue for this debate with RN Raw and Dr. Mick Leroy. So that's honestly going to be a blast. I'm pumped for it. But yes. Erica, do you live in Texas? You have to go.
I don't. I know. I think being in the UK for most of the year means I'm a little bit out of that range. But I would love to come. Hey James, come to the UK. Let's do a debate there.
Let's do a debate tour in the UK.
I'm actually, no joke. There's I was thinking about it earlier today of whether or not they could actually happen. And it's not.
Oh, that would be so cool.
It's not impossible. So it's definitely a possibility.
Hey, if you do that, I'll take you out and get you a drink at one of my favorite pubs.
That's nice of you. I appreciate that. And someone just asked in the audience, are there tickets? We don't have tickets. It's going to be a free first come first serve. It's actually going to be in a tattoo parlor. So we're kind of doing almost like the Bible and Beer Consortium. They go to pubs. We will do it in this tattoo parlor, and it's going to be a lot of fun. So I'll let you know the exact address as we get closer. And but yeah, so hopefully we can fit everybody in there, because I think there's going to be a lot of people. So it'll be a lot of fun. And next up, thanks for your super chat, Athena, Goddess of Wisdom and Warfare. They said, standing for truth is an all right guy. He's just wrong. What do you think of that, standing for truth? You like
Listen,
that?
There were some really, really good play on words regarding SFT. So, whoever can give the best play on words for SFT, I think what was one of them? Standing for Lies is a good one, but it's $25. No, I'm just kidding. But yeah, Athena, good guy. I've done a lot of discussions on his channel. I got nothing bad to say about him. I would say he's a nice guy, and he's wrong as well.
Gotcha, thanks for that. And so sorry about the sound. I played the boom button, because when I read that super chat, I was like, oh, good jab. And I played the boom button, and it was the most frightening, terrifying scream ever. And everybody's like, dear gosh, what was that? So thanks for your patience, guys. All right, Stupid Whore Energy, as she likes to be called, thanks for your super chat. She says, standing for truth, how do you explain the small population of Sumatran orangutans exhibiting high genetic diversity but thriving?
To be honest with you, I'd have to see the source and the paper on that one, but I bet some money that there's a pretty decent explanation for it, because it sounds like he's probably grasping at straws. But yeah, if he wants to send that, I'll give an answer afterwards. Never heard of it.
Gotcha. Appreciate that. And thanks so much for your super chat, Translator Carmium. Sorry, that's embarrassing. Okay, they say, standing for truth, how can you complain about a bottleneck of a few thousand if you believe in the post-flood Noatian bottleneck, which was much smaller still?
Good question. Good question. That's usually the question I get after I bring up the fact that their so-called population bottleneck is not even remotely feasible. But ours is very easy to explain. I mean, based on our model, okay, I'll take it slow for them, use really small words. We start with just two people. okay, constituting a very extreme yet benign population bottleneck, okay? Then, and you can just read this in the Bible, 10 generations later, a second single generation bottleneck of just what? Whoever knows their Bible, just 8 people occurred at the time of Noah. Both bottlenecks were very brief, just one generation, and then followed by explosive growth. These cases, there would be almost no previously accumulated mutations, especially if we explain the vast majority of DNA differences as functional created DNA differences, therefore no inbreeding effects. That's why the limited human genetic diversity we see is good for us, but a huge problem for evolutionary theory. And as you can see from the evolutionist explanations, it leads to unrestrained storytelling. So that's my answer.
Gotcha. Thank you very much. Next up, trying to move through fast, Superdora Energy, thanks for your super chat. She says, if mutations are degradative, why do viruses often evolve complex structures like HIV has evolved complex gated ion channels?
So is he talking about virus? So the HIV one, is he also referring to like endogenous retroviruses, like viruses in general?
I'm not sure if she only mentioned HIV.
You know what? I'd have to see, because I know the explanation for the function found in endogenous retroviruses is that these functions are co-opted, just like pseudogenes, for example, they're co-opted. But the thing is, that's just theoretical, right? We didn't actually observe these functions being co-opted, so I'd have to see if that's the same thing going on with the what's going on with the HIV as well.
Gotcha. Thank you very much. We appreciate that. And next up, Superdora Energy again, coming at you. She says it's been
He's busy.
That's right. She says it's been confirmed that that telomeres in nearby DNA undergo lots of mutations. So of course you'd expect this kind of DNA to be divergent and degenerate.
So she's talking about the telomeres. I guess she's admitting that the sequences that we should be seeing, if there was a chromosomal 2 fusion, right, we should see these remnant telomeres, and there's a struggle to find these because they're degenerate. So I guess she's admitting that they're degenerate, at least, which is kind of helps my case. But I would just ask, why were the telomeres even involved in the fusion to begin with? Their job is to prevent the fusion. So as far as I know, the telomeres don't occur in the middle of genes. Genes are not around telomeres. So I mean, there's so many discrepancies to the argument, to the story of the chromosomal 2 fusion. So
gotcha. Thank you very much. And then Erica, if you'd like to respond, I know Standing for Truth gets a lot of these, so if you want to like add something on some of these, like feel free.
I mean, I would just say on that we do have the remnants, and they're in Tompkins' own paper. So that's why I kind of like sassy pointed up at this. I was like, hey, there they are right there. That's all I have to say.
Gotcha. And we do have a question.
Hold on. If you have a question for Erica, perfect, because I got to run upstairs real quick.
We do have one.
Taking a 1 minute break.
Gotcha. Thank you very much. And so, Mitchell, thanks for your super chat. They said, question for Erica, if creation and evolution both include nested hierarchies, what mechanism naturally created your quote-unquote magic cell via abiogenesis?
Well, I think that's really a great question. I mean, basically, the field of abiogenesis is booming. I think it's kind of interesting that they asked that in a human evolution kind of chat, given that abiogenesis is entirely unique. But it all starts with chemicals, right? I mean, chemical reactions, they appear to have no real rhyme or reason to them. But as far as I know, it was amino acids that kind of began to clump together and sort of form these proto strands of RNA at some point, if you subscribe to the RNA world. That's on my field. I wish I could give you a better answer. Ask Jackson Weave. He knows this stuff.
You got it. Thank you very much. And I assume that Standing for Truth is still occupied, so I'm going to just quick mention a couple of things. First, stoked to have you here, folks. We rarely talk about the philosophy of the channel, but I think it's worth mentioning that we are basically trying to provide a nonpartisan platform, so we don't take any stances on the issues. We don't have after shows where we say who won or lost or anything like that. We are trying to be fully neutral and despite being imperfect, that's our goal, and we want you to feel welcome, whether you be Christian, atheist, Muslim, one of the many strange creatures in between all these different worldviews. We're glad you're here, and it sounds like, Standing, are you back?
I am back, my friend.
He's
All
back.
that tea and water will get to you after a couple hours.
Nasty. Thank you. So, very good. I appreciate that. And we do have a question.
No problem.
Don't worry. Stupid whore energy. Oh, nasty. So
she's putting your kids through college there, James, with all those super chats.
She does do a lot, and she says, "How much for James's snot-filled rag? I will pay." Nasty woman, very just disgraceful.
A lot of nastiness going on.
You've got some good questions.
Unfortunately, Steven Steen already offered $20. That's hard to beat. I mean, he's a nasty guy.
You're missing the big money here, my dude. You got to start capitalizing on these asks.
The snot rag. That's funny. Oh boy. Subtracted. Thanks for your super chat. They said, "Hey, Standing for Truth, why is the FOXP2 gene expressed in the chimpanzee genome as a mutation?"
What I know about the FOXP2 gene is it's the gene associated with language, but it does far more things than just that. So the fact that it's broken doesn't, give evidence that if it worked, they would. I'm not sure if I'm answering the question properly. Can you?
Yes. Let me pull it up 1 second. And I've got to undo this. Hey, Standing for Truth, why is the FOXP2 gene expressed in the chimpanzee genome as a mutation?
Okay, so it as a mutation. So if it was originally functional, like I said, it's associated with language, and if it does far more things than that, and now it's expressed as a mutation, I mean that would just be evidence of genetic entropy after the fall. I mean, some functional genes can be the result of mutation if that occurs.
Gotcha.
Okay, if I jump in real fast on that one, James, just a little quick anecdote. Since Standing's getting all the questions, why is no one asking me any questions? I'm bitter about it. But I think that's an interesting question. I'm not positive that the FOXP2 gene is language because I don't really know that gene particularly well. But if it is, the interesting thing is that chimpanzees have some like 86 or so unique gestures that they can combine to get across messages, and human infants display 46 of those between the ages of 1 and 2. So they do indeed have a sort of proto-language, and that can be extended to geladas, whose chittering follows Menzerath's law, or vervet monkeys, who have their own vocabulary for predators. So it is very interesting that we actually see a gradient of language, very proto-language, in living primates.
Gotcha.
Yeah, I think because just real quick.
Super
quick. I promise.
Okay.
Just like I said at the beginning, I just wanted to make sure I was answering it right.
Go on.
It can be associated with more things, more functions. For example, and I'll compare that to protein moonlighting, which back in the 40s, one gene, one protein, one function was the thinking. Now we know of protein moonlighting. These proteins are doing unimaginable things. It's like a multifunctional tool. So some of these genes have more than just one function. So it being a broken gene or a mutation is not, I don't think is going to convince me of ape human evolution.
Gotcha. Thank you very much. Next up, got one from Subtracted. Thanks for your super chat. They said, "Hey, Standing." Oh, we got that one. That's embarrassing. Okay. Stupid Whore Energy. Thanks for your super chat. She says, "Are you aware that two proteins don't need to have the same amino acid sequence to perform the same function?"
Is she referring to third position redundancy?
Let's see here. I'm not exactly sure.
Say it again.
Yeah, and you know what? A lot of those degenerate codons, for example, are also now, there's massive literature suggesting function. Because you can look to somebody like Dr. Herman Mays, who also looks at DNA level hierarchies, and he'll look at neutral variation, third position, degenerate codons. But now, even those are proving to be functional. They work to slow and speed processes in the cell. So it all falls in line with pre-existing heterozygosity. So yes, I am aware of that.
She followed that up with a quick one where she said, "Gene duplication is a very old concept." And she also said, "I love Nickelback." I don't know why I made that last part up.
I mean, the evolutionists will often look to gene duplication as a way to add novel information to the genome. But I mean, if I give you a copy of a book that you already own, and then I start introducing typographical errors into the second copy of the book you already own, now you just have a more messed up copy of the original book. So it's just more evidence for genetic degeneration. It comes down to net gain versus net loss. So that ain't gonna help.
Gotcha.
Quick add on that though. What about natural selection? Ooh, that's
right.
all
Natural selection, though, the problem is it can't act upon those near neutral mutations that are unselectable. Those are the ones that build up. Sure, it's going to amplify the best beneficials and get rid of the worst detrimentals. It's the near neutral mutations that accumulate and degenerate our information system. So that'll have to be the problem you'll have to address, Erica.
We should have a conversation about genetic entropy. I've heard you speak to like CRISPR and stuff like that before, and I would be very interested to have that chat. I'm not very, well versed, as I've said, for like the 20th time. I'm not very well versed in genetics, but still, it would be an interesting chat.
Yeah, I think that'd be a blast. You've been fun to talk to. We can definitely set that up.
Sure. Sorry, James. We started talking again.
Oh, James, you still there?
No problemo. So I appreciate that. And I think that's a great idea. You guys ought to. And the next one comes from Stupid Whore Energy again. She's persistent. She says, how do you explain the fact that ERVs, or dead viruses, are in the same exact places in humans and chimps, but not other species?
So good question. Regarding ERVs, it comes down to the fact that are they really the result of ancient viruses? Because now we know that the evidence suggests that these endogenous retroviruses and these retrotransposons, for example, they're indeed functional, and not just functional, not just low levels of function, but these endogenous retroviruses, they have crucial functions. They help regulate genes and even determine cell types. So like I said earlier, the evolutionist now has to claim that they were co-opted, but that is just more fancy storytelling. So these are important DNA elements, just as we'd predict based on created genetic diversity.
Gotcha. Thanks so much. Next up, we got one from Cy Gart. Thanks, Dr. Gart, for your question and super chat. He says, inbreeding requires a much smaller population than 2,000. Chromosomes are different between species because most of the genes on Y are not essential.
Yeah, I don't really think that matters because if they're different or not, I mean, the fact is, if we're talking about human evolution, chimps and humans do have a Y chromosome that's very, very dissimilar. And if you want to look at the Y chromosome, well, they can be traced back, proving one paternal line. And some of these degenerate chromosomes regarding the Y chromosome is just more proof of genetic entropy, the crumbling genome. That includes the Y chromosome as well.
Gotcha. Let's see.
I'm very flattered that Cy watched this conversation, though. Cy, you're awesome. Love your stuff.
I like Cy. I hope you enjoyed it, Cy.
Yeah.
Gotcha. Thanks so much. And by the way, so we are trying to, I'll mention that 99% of you all here are honestly so terrific, friendly, and you like to debate, but you're not like, like being like abusive, which is great. You're just superb.
Terrific.
And there's maybe like 1% that sometimes is a little bit like more fighty and a little bit more aggressive. And so in that case, just want to ask if you would be so kind. We're trying to like change the ever-changing culture, trying to mold it into like, yeah, we're going to try to only ban hate speech. But we do want to have the mods hopefully just say like, hey, like if somebody's being abusive, like, hey, can you do us a favor and not call somebody an idiot, something like that. So we're still going to try to like not regulate people. We don't want to like control people, but at the same time, we want to like kind of naturally, organically, like through your own free will, foster that kind of spirit of like kind of like warmth or friendliness. You know what I mean? Does that make sense? So
James, that's so diplomatic. That was such a diplomatic way of saying, cut that out now.
That's funny. My hope is that hopefully it'll just be, and like I said, 99% of the people, very kind, like it never are abusive. Once in a while, though, it's kind of like, kind of. But
yeah.
So with that, next up, we're going to try to fly through as many. We just got through the super chats, so thanks so much for those, folks. And we are going to jump into the other questions. I will try to do this as fast as possible. We're not going to get through all of them. I'm really sorry about that, everybody. There's a lot, and I want to respect the debaters' time. We've already...
I just have to jump in, James. To be honest with you, I've only got about 2 more minutes, so if I'm guessing most of the questions are for me, so you can go through him as much as possible. But
yeah, I know what you mean. I know what you mean. We've gone over your time, and I'm sorry about that, you guys.
I'm sorry.
No, it's been a blast. It's been a blast.
Don't worry about it.
And
All
so,
good.
Love your channel, James. You put on the best debates.
We usually try to do like an hour and a half, 2 hours tonight, but didn't quite do that tonight.
I lied.
So
hopefully it was one to remember.
Yeah, absolutely.
Honestly, it really was. People, honestly, there's just a lot of positive feedback. People have really been enjoying this and engaged, and so I've definitely enjoyed it. And we'll try to fly through just maybe 1 or 2 of these. Actually, what we'll do is maybe like 1 or 2 for standing, and then we'll do 1 or 2 for Erica, because that way standing you can go. And so 1 for standing, we will do this one from Rayward. This came in early. They said, I already know standing for truth will evade. Can you describe exactly what a transitional form between apes and humans would look like if you were to be convinced evolution was true?
I'm not the one. If you'll actually hear me rarely even bring up transitional forms, because when it comes to transitional forms, I'm not difficult with it. I think a transitional form would be the blending of 2 separate creatures. I mean, if you look at something like maybe Australopithecus sediba or habilis, or even the Piltdown man back in the day, these so-called human ape ancestors that are clear evidence of the mixing of Homo and ape bones. I mean, those would be probably close to the perfect transition. But as I've explained many times, when it comes to transitional forms, our model too predicts the mosaic, the transitional form. For example, I point to products of human design that seem to span 2 categories, just like you can find certain animals. Remember, the majority proves the rule. They can point to some, say, reptile mammal transitions, or they can point to tiktaalik, for example. It's going to need to be the rule, these so-called mosaics. But an amphibious assault vehicle, to the evolutionist, would be the perfect transitional form. So it would have to be a blending or a combination of 2 categories, right? A really good combination of human features and a really good combination of ape features.
Gotcha.
James, would it be okay if I said something super quick?
Yes.
Just like mega quick.
Correct.
I would compare that to being like, okay, so we have Archaeopteryx, and then the human, comparison to that would be an airplane with wings that doesn't fly and rolls around like a car exclusively, which we don't see. So, transitional forms are kind of indicative of kind of rigging existing structures. It would be all I would say about that.
Gotcha. Let's see. We will, standing for truth, we can cut you loose. We know you got to go, so we appreciate you being here, and thanks again for your patience. I know we went over the time that we promised.
I was wrong. I'm sorry.
It was a blast.
It was all a mistake.
Really enjoyed this debate. Good questions from the audience, too.
Yeah, absolutely. Thanks for chatting with me, Standing. I had a great time.
And
thanks so much, Erica. Hopefully we can do it again soon.
Yeah, absolutely.
It was a lot of fun.
Sure.
Quick.
We'll email. We can talk about it when I send you all those Crete papers.
Sounds good. I'm looking forward to it. Thanks.
All right. Great.
Thanks for doing it, Erica, and James, thanks for setting it up.
Thanks, Standing. Take care.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button, because we are just getting started.
Ladies and gentlemen, thrilled to have you here for another epic debate, as today we have Erica and Standing for Truth joining us for round 2. It's going to be an epic one, folks, and want to say, if it's your first time here, consider hitting that subscribe button, as we've got a lot more debates coming up. In fact, you'll see at the bottom right of your screen, Endgame, the final chapter between Mark Drisdale and Kent Hovind, will be this coming Tuesday. So our last debate between those 2 gentlemen, as it's always been fun to watch them. And so that's one you don't want to miss. You can set a reminder. So with that, want to let you know we are very thankful to have our guests here. In fact, not only that, but we're also giving a special thanks to Erica. She's a night owl, but nonetheless, it's impressive. She's over in London. Is it London right now, right?
Yeah. You can see my change of scenery. I'm in my normal living quarters. This is my flat, so I've got all the decorations up. Anybody who's subscribed to my channel, this is a familiar scene. So got the whales right here. It's gonna be great. I'm ready.
Epic. Very exciting. So it's going to be a fun one tonight, folks. We are really looking forward to it. I want to let you know what we are going to do for format. I'm going to pull up on email because I'm trying to remember, unless one of you remembers. But
yeah, we're doing 5 and 5 short intros and then open discussion. Yeah, standing.
Very good memory, Erica.
Hey, that's what I do.
Gotcha. Perfect. So it'll be, if I understand right, 5 rounds of 5 minutes. Is that what you're saying?
I mean, I thought it was openers and then open discussion.
Right. Yeah. No. It's
gotcha.
5-minute introductions and then just open discussion and then
Yeah.
Q&A. I'm guessing.
Thanks so much. Appreciate it. Long day already.
That's the fun way to do it, anyways.
You
That's
betcha.
what we're here for, James.
Stoked for it. Thanks for that refresher. And want to let you know, folks, if you have any questions, fire them into the old live chat. I will pull those questions out of the live chat, and we'll ask as many as we can at the end of that open discussion section when we get into Q&A. If you want to do super chat, that's an option as well, in which case it'll push your question to the top of the list for the Q&A, and it allows you to make a comment toward one of the speakers. Of course, they'd get to respond to it, and we ask you to be your regular friendly selves for that. And so with that, very excited, folks. I am going to set the timer for 5 minutes. And my presumption, because usually it's just the habit, we always have the creation person go first. Standing for truth, do you want to go first, or did you guys already decide together that Erica would go first?
No.
I don't think we didn't have a choice, but I don't care.
Yeah, it doesn't matter to me. I mean, to be fair to Erica, she started first last time, so I don't mind starting first this time, but ultimately it's up to you.
Yeah, sure. You can start. Yeah, I always start. You start.
You bet.
Only fair.
Okay, let's timer set. Standing for truth.
Awesome. Well, thanks so much. Thanks so much for doing this, Erica. The first debate was a blast, so I'm really excited for this one.
And
James, as always, brother, thanks for setting it up. So before we get started, we all must realize that it's genetics. It's in our DNA where the question regarding ancestry is answered. Genes, traits, genetics is what's inherited sperm and egg. Sperm and egg don't pass on a fossil or a bone. They don't pass on geography or a rock. They pass on genetics and traits. And so if we want to find out the history of humanity, this is where we must look. We have clocks in our DNA, for example, that go back just 6,000 years to Adam and Eve. Let's discuss some reasons why, based on genetic data, the Bible's account of human history is true. When we focus on the variation, for example, in the Y chromosome in the world today, we see that Y chromosomes have very little variation. This indicates that we came from a common ancestor in the not so distant past. Interestingly, the Y chromosomes are geographically specific. There exists African Y chromosomes, British Y chromosomes, Chinese Y chromosomes. Why is this? This is because mutations have occurred in the lines after Babel, since God divided the nations based on their Y chromosome. The family lines are based on the male ancestry. These nations were divided across the planet. Is there good evidence for limited ancestry? Or does the evidence tell us that humans did not go back to Adam and Eve, but are actually related to apes, dogs, and banana plants? Well, the evidence for the existence of Adam and Eve and limited ancestry is you and me and the genetics of the people in the world today. The genetic structure in humans today speaks to us of a literal Adam and Eve. We have a small DNA compartment called the mitochondrial DNA that we only inherit from our mothers. Everybody on this planet got their mitochondrial DNA from a single woman, who is exactly what we would expect from a biblical Eve. She is quite literally the mother of us all. In a nutshell, we have nearly identical mitochondrial DNA, and the very few mutations that differ from Eve's sequence are exactly what would be expected if Eve arose just several thousands of years ago. As I briefly touched on earlier, the same thing goes for the male side. Science tells us that all men have similar Y chromosomes, which is passed down from their fathers. And all of male humanity traces back to a single man. This single male ancestor of humanity has all the qualities expected if the Bible were true. The main argument offered, though, by the evolutionist community, especially biologos, against a literal Adam and Eve and ultimately limited ancestry has to do with genetic diversity. They will say there exists no real scientific way for two people to generate all the genetic diversity we see in humans today. This main argument is easily refuted and actually reflects a poor understanding of even basic genetics. A man and a woman today could have thousands, even millions of children, and no two children would be 100% identical. This is because they are heterozygous. There exists built-in diversity in millions of sites within the genome of the average person. In the limited time that I have here, the best way I can explain it is if God created Adam and Eve to be heterozygous, which in fact would be optimal design, you could have as much human diversity and more than we have today in their first family. Time is not required for the diversity today because diversity can be designed and built into Adam and Eve. This argument is not even scientifically reasonable. One more line of evidence I do want to point out before we go into the discussion has to do with the HapMap data. The HapMap project is what I am referring to. And this very important project was intended to compile a significant fraction of human genetic diversity. They examined millions of variants in thousands of people from around the world and made the data easily available. Dr. Robert Carter puts it perfectly when he states, and I quote, "I am quite happy with what we learned from the project." What have we learned? The human genome is young. Shared blocks of DNA are large, and there has not been enough time to scramble them to randomness. The human population came from a single source. Most blocks are shared among all world populations. The human genome is falling apart. Deletions tend to not be shared among populations, but are unique to subpopulations. This is further evidence for the youth of the genome, and that we came from a single source population in the recent past. I've got 10 seconds here. Genetic entropy, orphan genes, the chimp Y chromosome, endogenous retroviruses, linkage blocks, for example. There's many different lines of evidence we can look to that point to limited ancestry. And I'd be more than happy to discuss any one of these topics with Erica and show her why limited ancestry is true and universal common ancestry is false. Thanks so much.
Thank you very much, Standing for Truth. Just several seconds over. We'll give Erica the same leniency. So with that, we will jump over to Erica's opening statement. Thanks again for being here, Erica, especially given how late it is. It's a pleasure to have you.
Please don't worry about it. I will, if that's okay, share my screen. I know I normally have my nice monitor up, but I don't have anywhere to put it. So
happy to do that.
If that's cool, it's just a PowerPoint. I've never done the screen share thing, so bear with me, you guys.
Totally.
Here. All right, share my screen, and then can you guys see that?
It's now showing. So let me just squeeze it down to make it fit.
And I'll do one of those. Does that work? Okay. Very cool.
Almost there. 3, 2, 1. Okay, and we're set.
All right. So biology supports a universal common ancestry. This is the argument that I am going to make today. Now, generally speaking, when we look at a universal common ancestor, what we're kind of referring to is that all life on Earth shares a common ancestor in the form of a single-celled organism some 3.8 billion years ago. Now, as Standing mentioned previously, his argument is somewhat different in that he's sort of going for the creation orchard, so to speak. So if you can see in this picture here, this would be Standing's creation orchard on the left, where we have several sort of nodes that are acting as creation origin kinds, right? And the diversity within those kinds is stringent and kept separate from one another. Now, of course, my position would be the conventional tree of life, which is here on the right. So the question is, why do all of the fields in biology agree that universal common ancestry is indeed sort of the reality that we live in? And The reason is because, at least in my opinion, in my humble opinion, it is the case. This is, in fact, our reality. We can look to genetic support, we can look to paleontologic support, and we can look to morphologic support. Now, as Standing said, we're probably going to be talking mostly about genetics today. So while I included some slides here on paleontology and morphology, which I will skim if I have the time, we're mostly going to be considering genetics. So I've harped on this many times before in other debates and in other conversations, but genetics by definition dictates relatedness. This is simply the case. The same sequencing that links humans to all other humans links us to the other apes, all the mammals, all the chordates, etc. So when you look in this picture, again, as I've said multiple times before, the same logic, the same genetic sequencing, the same means by which we develop phylogenetic trees, Standing would agree within sort of the kinds. Like they, Christians tend to use kinds, sort of structure their kinds with genetics as well. The question is, can the lines cross? And my argument here is that there is no empirical reason to suggest that they would. There just isn't. It's a smooth gradient. And I've said this before too, but the sort of the same process that we use to allow paternity tests to be considered admissible in court. We use to reach the 98.8% similarity between us and the members of genus Pan, and then indeed 92 with the macaques and so on and so forth. So in addition, we could talk about endogenous retroviruses. Now here I've linked a couple of nice papers that cover the similarities of the ERVs between us and the chimps. And we have this nice little quote on the side that you can read if you like, which, to summarize it, 99.9% of ERVs in humans are at the identical location and are structured identically between us and the chimps. And even when we look at these specific ERV insertions, we find that the mutations that those ERVs experience in and of themselves are also identical, which means either we're dealing with a completely separate event for every single ERV in every single primate lineage, both within and without or outside of the human and the ape kind, and I'm the monkey kind, I'm not sure how Standing structures it, or we're dealing with a common ancestor. So my question would be, why can creationists not draw these lines between the kinds? Why do we see this smooth gradient that I've been sort of alluding to? You can sort of see the same thing when we look into deep homology, which would be kind of the argument that we're dealing with the same building blocks. So geology and paleontology, of course, continued to confirm a common ancestry, and not just that, they preclude a 6,000 to 10,000 year old Earth. Now, I know it might seem sort of tangential to bring this up in a conversation about genetics, but in genetics, we get sort of divergence times using molecular clocks. And interestingly enough, radiometric dating, a completely isolated physics-based sort of just equation that we use to measure radiometric or radioactive decay, confirms this. They corroborate one another. And not only that, but paleontological evidence can also be corroborated with other methods like ice cores or dendrochronology or natural nuclear reactors like the Oklo reactor. Then, of course, there's morphology, atavisms, vestigial structures, deep homology. Here are some nice pictures. Sometimes we see horses, display these additional toes that they used to have when back with Eohippus and the like. Vestigial pelvises, which I know some people say have a function in copulation, but not so. Ask any of the whales that require a third party to mate. We see deep homology in the form of the PAX6I gene. Rector pili with our hair standing on end, and of course our wisdom teeth. None of these make sense when we're sort of taking this idea of a designer into our mind. And I know that genetic entropy is going to be brought up, and that's the reason why we have these sort of degenerate traits, but I'm ready to have a conversation about that. So my question for Standing is sort of threefold. Well, I think there might be more than three, but where and more specifically, why do you draw the lines between the kinds, and what separates the humankind from the ape kind? You can do this genetically or otherwise, I don't care. Another one is, how can limited ancestry account for the gradient of forms that bridge the kinds in the fossil record? So here we have an amphicyanid, and we have a wolf, and we have a grizzly bear. Now, morphologically, it essentially looks like you took 50% of the traits from the amphicyanid and then sort of parceled them out into these two very closely related animals that we have today. And lastly, why do we see atavisms and vestiges in nearly every animal? And why do these vestiges seem to indicate and suggest that they're from our genetically derived ancestors. Why do they match evolutionary theory? What's the deal with a whale having a pelvis, and then also us finding similarly structured organisms living in the same location with the involucrum, which is an inner ear structure unique exclusively to the cetaceans, and through geologic time. So these are sort of my questions for the conversation, and that's it.
Thank you very much, Erica. So now we will switch back over to, or I should say, we'll switch into open discussion section. So I will close your share screen.
Thank you. I know how to do it. I'm hopeless.
Gotcha. Okay, thank you. Very sassy. I appreciate that. Next, we will go into the open discussion section. So, very exciting, folks. And the floor is yours. Thanks for being here again.
Yeah.
Awesome.
All right, Stevie, where do you want to start? You get to pick because that's how we did it last time.
Sure. Awesome. Well, I appreciate your opening. Thanks for the visuals. Definitely lots of good questions, lots of good topics. Since you started off with ancestry anyways and where we can draw the line and kind of how there's a perfect gradient, a perfect nested hierarchy of life, I guess you can put it. My question to you then would be regarding certain taxonomically restricted genes that we can look to separate ancestry, right? So if we're going to look at limited ancestry, for example, if we look at orphan genes, and orphan genes, they lack similar counterparts in other organisms, as in like there's no evidence for common ancestry with, say, humans and chimps because of these taxonomically restricted genes that are specific to humans and then also very specific to, say, chimpanzees or like the cat family. Or
well, sure, of course there would be, because the divergence time between humans and chimpanzees, the divergent time would be approximately 13-ish million years ago, with the speciation time being 6 to 7, give or take. So that's plenty of time for you to get emergence. That's of sort of genetic novelties. That's why we have ERVs that are also unique to humans. But you can point to those, and the exception does not make the rule. You can't point to the exception and say, oh, look, there are, individual ERVs or orphan genes, et cetera. I don't know nearly as much about orphan genes as I do about ERVs. But then I will simply turn and point to the 99.9% of ERVs that we share that are identical and in identical locations. So are you suggesting, just to kind of turn that into a question, are you suggesting to me that there have been separate genetic events regarding sort of these ancestral viruses for every single primate lineage, and then also humans, who I'm assuming you wouldn't accept our primates?
So good questions. I definitely want to talk about the endogenous retroviruses, but going back to the orphan genes first, as a counter to that, the orphan genes, for example, they're fully functional gene sequences, and what they do is they code for both a functional messenger RNA and a protein, okay? These orphan genes, though, Erica, they don't fit the hypothetical fairy tale of evolutionary theory, and our prediction based on ancestry and proving limited ancestry would be function, okay? So if these orphan genes, which according to our model are needed for the uniqueness that's associated with each created kind, as, say, described in the book of Genesis, which obviously we look to as a starting point, the fact that this amazing category of genes are taxonomically restricted and extremely functional and unique to each type of organism would suggest a limited ancestry for the main fact that they are fully functional. I mean, how did these come about in separate ancestries according to the evolutionary theory? I want to just make an analogy real quick, just for the audience. It's like looking at 3 different books and then finding specific words that have important meanings in the different books that aren't found in all 3 books, right? There's no evidence for any type of ancestry. That's why they call it orphan genes, right? There's no actual evidence for the ancestral genes leading to these genes. So I mean, how would those pop up in evolutionary theory?
Well, I'm asking though, why are you yes, things can be taxonomically restricted. I mean, obviously there are, as I said previously, there are sort of unique ERVs to humans. There's also unique breakages like that are just in the primate order. There's a breakage that does the same thing, the gulo breakage, that also exists in guinea pigs, but it's very different. It's in a different location, and it lacks some of the sort of regulators that ours has. So I'm kind of wondering why it is that you're suggesting that in any shape, form, or fashion that these orphan genes cannot emerge after splits, after divergence points.
Well, I mean, it can be suggested that they do pop up. Like, I believe that the evolutionist explanation is de novo gene synthesis, that they popped up through the non-coding regions of our genome. But the question is there any empirical observational data for that occurring? And the fact that they've popped up to be extremely functional in the genome seems to be more ad hoc, post hoc reasoning. But if I'm arguing for limited ancestry and these types of taxonomically restricted genes that are extremely important in the genome, or what are expected to be found, and this is exactly what is found.
Right, but what you're doing,
this would suggest limited ancestry. It's exactly what we expect to find. Go ahead.
Right, but you've got to. I think a clear distinction has to be made. So when we're looking at data in general and we're trying to support a hypothesis or see if the data does support a particular hypothesis, it can. Obviously, as we know in the nature of science, data can support multiple hypotheses. But the sort of the issue with this is you're suggesting the orphan genes. Again, that's kind of why I wanted to make it more about biology in general, because I'm not a geneticist, but I know enough about it. Yeah, but I know enough about genetics to know that these kinds of sort of emergent genes, ERVs, et cetera, things like that, they don't preclude evolutionary theory in any way. You're arguing that it supports limited ancestry. And again, I don't know enough about it. It may support limited ancestry, but it also doesn't preclude evolutionary theory and universal common ancestry. And in light of pretty much, in my opinion, everything else that sort of I brought up in my presentation that I'm sure we'll touch on later, which not only does it support universal common ancestry, but it also precludes limited common ancestry. You see what I'm saying? Because there can be data that's like, yeah, it fits both frames. Like I know Jensen likes to do that with nested hierarchies, which I disagree. But the fact of the matter is then he turns, and I'm sure we'll do the same thing, and covers other subjects that he believes supports limited ancestry by precluding universal ancestry. Well, right, so it sounds to me, you get what I'm saying. Sorry.
Right, yeah. And if we look to homology, right, engineers build in a homologous type pattern, or we can look at DNA level nested hierarchies. We can look at nested hierarchies on an amino acid level, right? I know Dr. Herman Mays likes to look at neutral variation, for example, but when there are competing hypotheses, we need to look at lines of evidence that can differentiate between the two. Now, I disagree.
Absolutely.
Right, but I disagree with your point in that orphan genes can be explained through fish to fisherman evolution because of the important functional roles that have been discovered in them. They are taxonomically restricted, and I've yet to hear an explanation from you as to how these orphan genes can pop up, can be specific to lineages, for example, humans, to the great ape family, to the cat family, to the dog family. How are these popping up and having very important functional roles? I mean, that doesn't just happen. We also don't see that empirically in the lab. That's the thing. There has to be some type of explanation in order to have that competing hypotheses, but I don't see no explanation for these. That's why they call them orphan genes. That's it. You know, sure. They belong in specific taxons, and that's it.
Sure, but you're suggesting that the existence of, and I know that you mentioned that sort of the conventional science side of it would say, okay, well, it's kind of this de novo emergence, but that does happen. I mean, I know you're frequently a fan of using ERVs that play roles in embryologic development because they've sort of been retrofitted. That's like a huge tenet of evolution, the atavisms, right? Like this idea of taking, something that used to do something else and, or not atavism, sorry, in some cases of vestiges, and using it for something new. I mean, because you're right. Most ERVs, though, have zero function, but some of them do. And they did pop up, right? At least according to
real quick before you get, I understand that you want to move on to the endogenous retroviruses. Just to kind of end the part on orphan genes, we can do that. I don't want to beat a dead horse. Let's move on to endogenous retroviruses and retrotransposons, for example. I would just disagree that orphan genes aren't a line of evidence that can differentiate between the two hypotheses, right? Universal common ancestry and limited ancestry. We can just agree to disagree.
And we
can. If you want to look to the de novo gene synthesis, we definitely need to see empirical data, whether it's in the lab or some type of technical paper that can actually show a non-coding region of our genome suddenly pop up and become functional as a protein coding gene.
So I kind of know where this is going. So I am going to openly, in front of everyone, make sure that this is clear. I don't know very much. In fact, I know very little about orphan genes. What I do know quite a bit about, or a decent amount about, is endogenous retroviruses. And the reason why I'm making the connection is because it sounds like conceptually there are quite a few similarities. So I'm not saying that I know enough about orphan genes to say this is certainly the case. I probably need to look into it, but I don't particularly find presenting a piece of evidence that, knowing absolutely nothing about it except for the blurb that you've given me here, could go either way in light of everything else that we're getting ready to discuss, convincing as a gotcha. You know what I mean? Because it sounds very similar.
There's many lines. Yeah, no, I agree.
It sounds similar to the gotchas that creationists use with ERVs. That's why I keep making the comparison. Not because I'm trying to dodge away, I don't want anybody to know I don't know very much about orphan genes. I don't. I'm not a geneticist, but that's the way the cookie crumbles sometimes. So I'm just saying I don't find that convincing. I mean, if there are, geneticists out there who are saying, yes, de novo orphan genes can happen, then the fact that we have de novo ERVs, emerging from ancient viruses and such things like this, and of course, new information through various mutations, I don't see a problem. It's completely in line with other conventional arguments that we're going to talk about.
Right. If I could just jump in real quick, because I definitely want to jump into the ERVs and the retrotransposons, and I'll give an answer to that. Just on the orphan genes and in the, it's not necessarily just a gotcha type argument. I will go on record that, the evolutionist community, they are somewhat baffled, and that is why they look to de novo gene synthesis. But to me, that's more of a circular argument, because the evolutionists know that orphan genes exist. Therefore, they need to hypothesize, why do they exist? So they resort to de novo gene synthesis. Well, why do they resort to de novo gene synthesis? Well, because orphan genes exist. So I'm just saying it's more of a circular based argument. Now, when it comes to the co-option in the functions found in endogenous retroviruses, I agree that these endogenous retroviruses, or the retrotransposons, for example, Erica, they are shared among humans, among some of the apes, of course. You can find nested hierarchies in the placement and positions of the ERV. So I agree with that. But the question I think we need to ask ourselves, Erica, is what are these endogenous retroviruses? What are these retrotransposons? Are they actually the leftover remnants of viral infections in our DNA, like you're suggesting, right? Or are they, say, created units of DNA function? Because for years, Erica, it has been assumed that they were the former, right? The leftover remnants of viral infections. And that is because, like you iterated in your opening, they bear strong similarities to actual viruses. And this predicts that these sequences are non-functional. And I know you look to co-option as an answer to the function, but this is a testable prediction based on the design model, and we are beginning to accumulate data. So what we do know at this point in time, Erica, is the trajectory of experimental results, and the trajectory points to increasing and surprising levels of function. So to me, it looks as if these are not remnants of viral infections, but actual functional DNA units. Now,
Right.
what's their
So
role in ancestry and in speciation? But this specific prediction, it does differ from creation and evolution expectations. Go ahead, take as much time as you need, Eric.
sure. So I'm going to kind of respond to that in twofold. One, I'm not kidding here. We do know of a lot of endogenous retroviruses. We're not completely in the dark about this, and I know of like a handful that actually do something. And so I'm kind of wondering where you're coming with this sort of concept of, oh well, the trajectory is we're going to find out that they have more function, and I think I know where you're going with it, and I think it has to do with the ENCODE project, because I've heard you speak sort of before on ENCODE, and for those of you out there who don't know, ENCODE was a sort of group that did quite a bit of genetic work on discovering the functionality of the human genome, and they came out with this quite large number, 80%. They came out to say the human genome was functional. Now, their definition of that sounds impressive until you actually read the work.
Erica, I don't want to interrupt, but I feel like ENCODE in general is. I just feel like ENCODE at this point is kind of a rabbit trail, because we're specifically on the endogenous retroviruses.
Sure, but you'll see the point I'm going to make. It's relevant.
Okay.
Because I actually watched your discussion with your series of discussions with CRISPR earlier today. And you mentioned several times this concept of, oh, well, the trajectory seems like we're going to find a lot of functions. But so I thought to myself, okay, well, time to look into that. Let's see, because I know CRISPR was saying, no, absolutely not. The vast majority of people in the field are saying, similar to ERVs, no, it's not looking like we're going to find more function, because every time they investigate, they find a function for something new. It makes up a smaller and smaller and smaller proportion of the genome. And there was a paper that came out in 2017, very recently, that put an upper cap and very unlikely upper cap of 25% of functionality, with a likely range of 10 to 12%. So that's the most recent literature on that, and it doesn't follow the trajectory that you were mentioning. So I don't think that you can say, because you find functions in one ERV, or you find a little bit of function in say, an intron or whatever, you can't just say the trajectory seems like we're going to find more and more, and then eventually we find out all ERVs are functional, or all of the human genome is functional. That's all I had to say. That was the connection.
Oh, awesome. And I appreciate that response and those points as well. And I did enjoy my discussions with CRISPR, and would definitely recommend anybody watch them. But you say that like just one endogenous retrovirus, for example, was found to be functional.
Oh, that's hyperbolic. I'm sure it's more than one, but continue.
Okay. No problem. Even in the ERVs, though, it's not just low levels of function, and that's the problem that the evolutionist faces, because the ERVs, they actually have crucial functions, Erica. For one, they help regulate genes. So they're not just functional in the embryo. They help regulate genes and even determine cell types. So I know that you're claiming that they were co-opted, but I would just say that that's mere uninformative gloss, because I would have to see empirical evidence, whether it's in the lab or some type of technical paper, that actually shows us a non-functional endogenous retrovirus going from non-functional to extremely functional in determining cell types or regulating genes. Before I continue, actually, would you be able to present something like that?
Hold on. What I want to talk about is your request there. You're saying, okay, well, I want to see.
I'm not saying you have to present it right now, but does something like that exist, where it has been observed empirically that a non-functional endogenous retrovirus went from non-functional to a functional DNA element, important in the embryo, important in regulating genes, important in determining cell types? I mean, could you present that to us?
I mean, I would say you've already done so, because we have, what I mentioned earlier, right, is that we have several, and what you agreed to is that we have, so many non-functional ERVs, you can't hardly list them out on a piece of paper. And then we proceed to talk about a functional ERV. So I think the burden of proof is on you to find function for them. If the vast majority is non-functional, and you're making the claim that functions do exist for all, or at least the vast majority, the burden of proof is on you to show that they have function, not on me because we have point A and point B.
Well, real quick.
This would be
Because you
like
brought up
finding.
well, Rook, I don't want to, because you brought up some things about ENCODE. I was kind of just clarifying with you. And I want to kind of touch on everything, because it's not just one or two ERVs that are being found. I mean, there's accumulating paper after paper. I mean, I've got papers here from 2019 that are now showing these ERVs. They frequently act to distribute regulatory information and thus confer genes with new patterns of expression and function. The
Standing, I don't
transposable
doubt. I
elements.
don't doubt it. I don't doubt that there are tons of ERVs that have functions.
There's
The
no
problem is,
exact-
and I checked today, the vast majority of ERVs are functionless. And
Have
so
they
because that,
have they functionally tested the vast majority of endogenous retroviruses, though? Because remember, no, real quick, evolutionists assume, and you admitted it earlier, that the vast majority of our genome is based on genomic fossils, evolutionary leftovers. But far from being junk DNA, the pervasive retrotransposons or the endogenous retroviruses, for example, that populate our genome, they have a powerful capacity to influence genes and even chromatin. I mean, the functions are endless. I can go on
yes,
and on. Even
some
the
of them
pseudogenes.
do.
We're
The way-
finding. Well,
the way-
I'm saying that your argument about co-option and suddenly the burden of proof is on me is more of a circular argument, just like with the orphan genes and de novo gene synthesis. So you're
It's
seeing
not-
that these real quick, I'm almost done. You're seeing that these endogenous retroviruses are playing very important functional roles in our genetics. So instead of actually giving us and providing us empirical evidence for the co-option of these endogenous retroviruses, you're just assuming that they've been co-opted because it doesn't fit the evolutionary theory, but it hasn't actually been demonstrated. And I'm saying that the trajectory of discovery does favor genome-wide functionality because
No.
every single month, more and more papers come out suggesting more and more function. That's
No,
why.
you're okay. First, I'm going to make a quick analogy. If I come out and I say horses are the most important mode of travel, that's just what they are. And you say, well, no, that can't be so. And I say, yeah, look at the Amish. They can't get around without horses. And then you say, well, no, because mostly people get around in cars. And then I say, no, it's really, really important in this one particular case that the Amish use horses.
Right, but
Are
it's
you
not just
getting
one
what I'm
particular
saying?
case.
These functional ERVs have indeed been co-opted. And the way that we know that they've been co-opted, other than the fact that they look very much like ancient viruses just in structure, when we look at them genomically, is the fact that the vast majority don't do anything. And when you say things like, well, the trajectory looks like, that's a very vague statement, because the fact of the matter is your talk with CRISPR was what, at least a year ago? And I checked today and they capped it right where he said they were going to cap it. He said upper limit, I think he might have said 20%. So he was a little off. He said upper limit, no more than 20%.
Right.
2017, upper limit 25%. And
real quick,
I'll pull something up for you.
I'm actually happy that you've at least admitted to that, because I even debated a biologist, I think it was 2 weeks ago, and I've debated like T-jump, for example. And every time I ask that question, right, how much of our genome, both non-coding DNA and protein coding DNA, do you believe the evidence suggests is functional? And you just said it there, just like CRISPR said, it can't be any more than 20%, 25%. But the thing is, that's because any more than that makes that type of fish to fisherman evolution impossible, simply based on mutation accumulation. Well, I've got a paper here that suggests that the genome has to be under 20% functional, or else ape-to-man evolution could not. Well, actually, real quick before we go on too many rabbit trails, because I still want to touch on ENCODE, because a few things that you said I disagree with. Are you disagreeing with the fact that we do see massive amounts of literature nearly every single month that gives us unexpected functions of RNA? And then as you answer that too, how much of our genetics, of our genome, do you believe we've functionally tested according to the gold standard of genome testing? Go ahead, take your time.
So first, we can swap papers in a moment, because I straight up sat down today. I sat my butt down on Google Scholar and I combed for all literature that I could find that was recent on functional DNA. And the most cited paper, the most seemingly sort of groundbreaking biggie that came out this paper in 2017. Now, of course, we're always discovering new things about genetics. And of course, some of those things are new functions. But again, this conversation you had with CRISPR was at least a year ago. And
Right.
we have discovered we haven't passed the cap. We haven't even gotten close to passing the cap that he set. These guys are saying, and I'm going to read this to you instead of sharing my screen just because it's annoying for everybody I know and it causes a delay.
Well, real quick
But
before you do that, can you just answer the question as to how much of our genome has been functionally tested? Because that's important in this discussion. Because
I'm going to respond to that with the same sort of rhetoric back and forth that you and CRISPR had. So humans are mammals. And that's why we use mice and that for testing and things like that, our drugs and sort of pharmaceuticals. And that's why our pharmaceuticals tend to work so well, because we share so much in common. We have done extensive knockout tests on mice. We can knock out so much of their genome and they will be just fine. They reproduce, they have babies, their babies are just fine. So that's because that section that we're knocking out is non-functional. It doesn't harm them to knock it out. So, because I know this ENCODE conversation is going to go down maybe the non-coding or the non-functional quote unquote DNA is actually for development. That debunks it right there. The fact that these mice with enormous portions of their non-functional DNA knocked out can go and reproduce just fine. They might have a couple of weird little morphologies, but other than that, their fitness is increased.
Before you go on for too long with the mice, the problem is many non-coding RNA genes, even pseudogenes, for example, much of the non-coding regions of our genome, they're only expressed under certain conditions. And much of the genome is likely involved, for example, and used between conception and birth. And the fact that many of these non-coding sections of our genome are only expressed under certain conditions, you'd need almost unlimited sensitivity in these genetic knockout tests being done on mice to conclude with any type of certainty that those genes being knocked out really are non-functional. Because if these genes are being turned on and off, let's say after the developmental stage or just based on environmental conditions during the life of the mice, especially because these mice, the genes are being knocked out in the laboratory. Therefore, many different environmental conditions that are outside of the laboratory, that a certain gene that isn't expressed and it's being knocked out, well, you're not going to be able to know that it's functional or non-functional. And the answer to your question based on genetic knockout tests, especially with humans, because it's unethical, it's less than 1% of the genome that's actually been functionally tested according to the gold standard of science. So I even asked that question too. I asked that question to CRISPR, and he went on looking for a paper to show how many of these experiments were performed based on these genes that are only expressed under certain conditions, and he never came back with the specifics on that. So you'd have to show that. I don't believe that those types of knockout experiments on mice have been performed.
To be fair, in that conversation, while he was looking, you were like, you can just get into me later. So there is always the possibility, as I frequently do, that he just forgot to send them to you. Now, I obviously have not,
and that's fine. And that's fine.
Yeah, and that's fine. But I feel like you kind of obfuscated the point there, because again, for everybody who's watching, right? These knockout tests have been done on adult mice, or rather the impacts are on adults. They reproduce, they pass those knocked out genes to their offspring, who develop just fine, and then reproduce themselves after living their merry mice life. So the point is that if you're suggesting that there is no plausible way that I can see to suggest that genes that have been knocked out, even though we've done it on enormous portions, are secretly doing some kind of function when nothing bad happens when you turn them off completely.
Well, no, I'm not saying
having a bunch of already broken breakers.
One sec, Well, just to be
I'm
sure
not
that
saying-
Erica's finished here, because we had
No, yeah,
you talked for a
it's
long
okay.
time, and I want to just be sure Erica gets plenty.
It's
Also,
all right.
you got it.
Well, I'm not saying that I'm not using a pseudoscientific argument suggesting that many non-coding RNA genes are only expressed under certain conditions. I mean, this is actually a fact. And my main point regarding that is we have only begun to discover just how functional, for one, the human genome is, and how functional the mice genome is. And I'm saying it's mind-boggling how many experiments, and I'm not saying it's impossible, but these number of experiments have not yet been done, and that's a fact. And
It's not though, because knockouts have happened, and the mice still reproduce.
Right. But the amount of experiments that would be required to conclude, is my point, with any level of confidence that a particular set of DNA sequences has absolutely no function, is mind-boggling. And
yeah, but the amount of experiments that you would have to do to conclude anything without beyond a shadow of a doubt is astronomical. The point is that the fact that you can knock out enormous portions of a mouse and then the mouse can reproduce and then its offspring can also reproduce because nothing of merit has been lost is parsimonious only with evolutionary theory, that's what I'm saying. To use your word from earlier, the trajectory indicates that enormous portions of the genome can indeed be knocked out because they don't do anything. So, and I want to segue this in because
I didn't say that they don't do anything. I'm saying a lot of those regions in the because the non-coding DNA, it functions much like a computer, okay? It's involved in regulatory functions. It's involved in embryonic development. It's even involved in the stagings of proteins for.
But they can't regulate if they're off.
But I'm
if
saying
you knock
that,
them out, they can't do anything.
No, but if these specific genes, and we have to look at which ones, what types of developmental windows, which types of environmental conditions, because if these experiments are being done strictly in the lab on these mice, well, these non-coding RNA genes, for example, even some of the pseudo genes, yeah, you're going to knock that out. You're going to snip it out, and it might not have some overall effect on genotype or phenotype, but that's because it's just not expressed.
Yeah,
so
but we're not talking about.
Just real quick, if you consider that, and then you consider, if you look at comprehensive knockout experiments for humans, for example, it's unethical, obviously, at this point. We functionally tested, genetics-wise, under 1% of the genome when you consider non-coding DNA and coding protein coding DNA. But the thing is, and the question I asked you earlier regarding the RNA, we do see massive amounts of literature that gives us unexpected functions of RNA. For example, are you familiar with protein moonlighting? Because
yes,
in the past,
of course.
Right. In the past, one of the ideas, you can just pick up a textbook from 40, 50 years ago.
One gene, one function, or protein, one function. Yes, I'm with you. Continue.
Exactly. Now, if we're looking at the protein multifunctionality, or we're looking at, say, on an amino acid level, these redundant codons, and I'm sure you've heard me say this before, those were always assumed to be absolutely neutral. Now we know they don't just tolerate error in the cell. These redundant elements, they're used to slow and speed the process. So the level of information compression into every single letter boggles the mind. It's actually incredible. And even
I'm sure that is indeed the case, but that's not the problem that I have with your statement that and in this conversation and in previous ones, that the majority of the genome is functional. The fact of the matter is, if you knock it out, it doesn't work. It doesn't do anything. If it doesn't do anything before you knock it out, there's no harm, no foul, right? So the fact of the matter is, if they do something important, anything, regulation, whatever, it doesn't matter. If they have a job that's even minute, if you knock it out, that job doesn't get done.
How much of the mice genome has been knocked out, though, on a percentage-wise?
Oh, I could not give you a number on that. If you
well, I could, and I could be corrected on this, but the last time I checked, it is less than 30%. So based on the fact that these tests are being done in the lab, based on the fact that less than 30% of the mice genome has been knocked out, and the fact that, as I've said, well, 3 or 4 times, many non-coding RNA genes are only expressed under certain conditions, less than 1% of the mice genome, the human genome, mammals in general, has been tested for genome functionality. And all you have to look at, like I talked about earlier, you just have to look at the history of genetics. Like you said, back in the 40s, one gene, one protein, one function. I've said that multiple times. You can see how many things have been changed. So yeah, I agree that the results from the ENCODE project, Erica, I do agree that based on their suggestion that at least 80% of the genome is functional biochemically, right? Because it's showing that the non-coding DNA is transcribed into RNA. Those are preliminary. I agree with that, but I'm saying the trajectory is clear based on genome testing, based on the history of genetics, that the trajectory does suggest most non-coding DNA in the human genome is functional. At least it serves in a regulatory capacity. I mean, do you actually stand by a genome of only 20% max functionality?
Here's the thing, Standing. I go by current literature. And what I try to do when I don't know something about a topic is I check the most recent literature, I see how many times it's been cited, and I see who's on the paper and investigate their background. Because that's, it's just like what they teach you. It's appropriate sourcing. And I'm going to read you this excerpt. It's quite short from this 2017 paper, which was New Limits to Functional Portion of Human Genome Reported. An evolutionary biologist at the University of Houston has published new calculations that indicate no more than 25% of the human genome is functional. This is a stark contrast to the suggestions made by the scientist with the ENCODE project that as much as 80% of the genome was indeed functional. They took a deceptively simple approach, basically, to sort of sussing this out by looking at deleterious mutation rates and replacement fertility rate, which is quite odd, and I wouldn't have thought to do it, but I'm also not a geneticist and that's not really my world. But this is the quote that I think is important. This is from the paper. It's Grauer et al. If you want to look it up.
I've got that in front of me.
Or I can send it to you, yeah, too.
I have it in front of me.
Oh, good. Okay. For 80% of the human genome to be functional, each couple in the world would have to beget an average of 15 children, and all but 2 would have to die or fail to reproduce, he wrote. If we use the upper bound for the deleterious mutation rate, which is at 2 × 10⁸ mutations per nucleotide per generation, then the number of children that each couple would have to maintain a constant population size would exceed the number of stars in the visible universe by 10 orders of magnitude. So this is the most recent literature. Not only does it say, yeah, probably no more than 25%, they're saying it's impossible. So that is evidence that precludes, it's just using math, deleterious mutations and reproductive rates, replacement rates and things like that. And it directly precludes what you just said about trajectory. Because like you said, these papers come out every day. And every time the new DNA that they find that's functional, the new portion of the genome, gets smaller and smaller and smaller. They might say, oh, wow, we found out that 0.15% of this, given portion is functional. Add it to the list. But it's sort of exponential. There's not that much more to kind of sort of complete what we know, at least about the proportion of what is and what isn't functional. I encourage you to read the paper because I'm not a genetics person, but I found it quite easy to understand. And the math is pretty straightforward.
Right. So I appreciate that response. So in that paper, what Gore says, he says that human evolution would be very problematic if the genome was 10% functional, but would be completely impossible if 25% or more was functional. So what he's demonstrating here is what results from an evolutionary mindset, because he knows based on the possible functionality of the genome, if we look at mutations, if we look at what's inherited, mutation accumulation in general, it would make that type of ape to man evolution impossible, because of the fact that if you have a genome that is, say, 50% functional, 60% functional, well, that means these so-called neutral mutations that supposedly build up with no real effect on phenotype and genotype would actually be more so nearly neutral.
Right.
That
So
means
you're saying there's a heavy bias, is what you're saying.
I'm saying there's a heavy bias, and you can even see that with the ENCODE researchers themselves. You can see that they did their research without that evolutionary bias. And evolutionists like the author here in this paper, they would have to fight the results of ENCODE. They would have to fight any type of trajectory that suggests the genome is any more than 20 or 25% functional based on the accumulation of mutations. Because, for example, Komura's theory of neutral mutations, right? Neutral mutations build up. Those neutral mutations are used over time by organisms to change, to adapt, to modify. But
well, Komura, come on, Komura was taken greatly out of context. I looked into that as well, with Jameson or Tompkins, I can't remember which one. But I want to make a very short, brief comment.
Real quick before you do go on to that, and you take as much time as you need. The point that I'm making here is obviously I disagree with the with the idea or with the belief that the genome is 20% functional or 25% non-functional or functional max. Because, and even if I were to just say concede that or just choose to agree to disagree, even if though 90% of the genome is perfectly neutral junk DNA, say only 10% of it is functional, that's still a huge problem, because based on what we know about the human mutation rate, right? We accumulate 100 new mutations per person per generation. That means, Erica, if I were to just agree with what you're saying, about 10 harmful mutations are still arising in every person, every generation. That means even the most fit individuals are still more mutant than their parents. And so even with intense selection, if we're looking at natural selection here, against the less fit, it cannot stop mutation accumulation. But like that paper suggests, the more functional the genome is, the more of a problem genetic degeneration becomes. Okay, take as much time as you need.
Yes, so here's the thing, and I'm going to use this to segue into genetic entropy, because I feel like that's kind of where you're pulling a little bit, which is fine, because I got my notes right here, because I don't know enough about genetics just off the top of my head, give at least numbers. But I want to make a quick comment about bias. Now, in the social sciences, anything that require, many areas of science that require an interpretation of the data, bias can be problematic. Here's where bias can't be problematic: with math. When you're just doing equations based off of either known rates or variable rates or whatever, as long as you've got your error bars, they're going to cover your bias. That's what they're there for, to say, okay, well, it's give or take, right? So you can say, yeah, he's biased to evolution, so's the vast majority of science. And I would agree with you, because I am also biased towards evolutionary theory, but it doesn't matter, because all he did was use math to get his point across. He plugged in equations. You can say I disagree with it, but that's the same thing as being like, I disagree with whatever, the Pythagorean theorem or something like that, just like an equation. Now, if you went in and you discovered, I guess that's a bad example. If I showed you an equation and you were like, I disagree with it. Now, if you go in there and you find an actual problem with it, that's another story. But as far as bias goes, it's not problematic when you're dealing with mathematics or physics or things like that. Or at least it's far less of a problem, because there's less room for interpretation. Now, as far as genetic entropy goes, it's not real. Genetic entropy, or at least the conventional science term, is error catastrophe. And it doesn't happen for several reasons. If you've got, go ahead and give me, I want to make sure that I'm kind of addressing the proper point, so I don't go on a big spiel and then we're talking about something different. When you talk about genetic entropy, are you talking about the accumulation of near neutral mutations that causes degeneration?
I would say if you look at genetic entropy or genetic degeneration, for example, it is the fact that natural selection cannot stop the accumulation of most harmful mutations. The problem is that the human mutation rate, for example, it is widely accepted to be 100 new mutations per person per generation. Now, even if I were to agree to disagree on your point or assertion that, you know, max functionality of the genome is say 20%, 25%, even though I want to make one last point about the function of the genome anyways, I would say that it is a fact that ENCODE, which you were referring to earlier, it does at least show that most of the human genome is functional, but also functional on many layers. Like we literally have layer upon layer upon layer of programming, Erica, within the genome. And no computer program has ever been seen to arise by trial and error or by chance. But genetic entropy would suggest that these mutations that evolutionists are saying drive the evolutionary process are either nearly neutral, which are slightly deleterious to the genetics, or detrimental. And that's the problem, is what type of selection is going to be able to filter out all those bad mutations, whether nearly neutral or just detrimental. Go ahead.
So the thing about ENCODE, again, just to give my, I guess, final point on it, for those of you who are watching, one, the definition of functionality is incredibly loose. And if you watch Stanning's conversation with CRISPR, who is has sort of hands in many different fields. He knows quite a few people who are, he's a cancer guy himself, but he knows quite a few people working in various areas of bioinformatics and things like that. And his conclusion was that he could maybe find 2 people out of the dozens upon dozens upon dozens that he works with who would agree that the definition of functionality was even remotely appropriate, because their definition of functionality covers everything. And the reason is because that's how you sell a project. I've been trying to sell my project for grants, because I'm doing a master's program right now, for several weeks, and you have to make it sound good or else you don't get the funding. So, and that I'm not saying that like it's anecdotal. That's how it is. That's it. That's just academia.
And just for the record, I'm not disagreeing with the definition that ENCODE used and the definition that CRISPR says that they use, right? Because ENCODE defined function as specific biochemical activity. And that's why I agree with you that the results suggesting that over 80% of the genome is actively transcribed in RNA is preliminary. That's why we've made a prediction as creationists that based on further genetic testing, knockout tests, for example, or testing mice under various types of environmental conditions where these non-coding RNA genes can be expressed in various ways is functional. That's our future prediction. And I think
yes, of course. And my answer to that would be ENCODE has already been busted by this 2017 paper. And I think there was another one in 2016, maybe, but I picked the most recent one because that good practice, I suppose.
Do you know how many, I think this is an important question before you move on. Do you know how many cell types ENCODE tested?
Oh, no, I don't.
Well, here's the problem. Here's the problem. So based on what ENCODE discovered regarding the function of our DNA, I understand that they defined it as specific biochemical activity. We have more than 1,000, and I'm sure you already know this, we have more than 1,000 cell types. So I'm saying, and I'm saying this to you in the audience, and to James, the moderator, I'm saying that more function is going to be discovered in the future. Because if you're testing only 200 cell types and we have over 1,000 cell types in our genetics, I think it's just based on an evolutionary bias to say that max functionality of our genome is 20 or 25%. And I know that the reason why he concluded that in the paper is based on mutation accumulation. Because if it's any more than that, mutation accumulation makes ape to man evolution impossible. That's a fact.
The problem though, is that I don't know, excuse me, I'm not sure for certain where you're getting your mutation rates from. I have a hunch that it's GeneSense rates, or at least Kimura, the people that GeneSense sort of pulled from. And if I'm correct on that, which I don't know that I am.
Are you saying 100? Are you saying the 100 new mutations per person per generation? I mean, you could,
No,
you could find that all over the place.
whichever mitochondrial rate, mutation rate, and nuclear mutation rate that he uses to make the case in replacing Darwin. Am I correct on that or no?
No, well, right now, I mean, if you want to get into replacing Darwin and the created heterozygosity hypothesis and mitochondrial DNA mutation rates and stuff, I mean, that's a totally different topic than
all I'm asking is it the same mutation rate that Jensen uses that you're using to say it over 25%, or not over 25%, that the deleterious mutation buildup would make ape to man evolution impossible?
No, that's just secular literature. I mean, the human mutation rate is generally known to be 60 to 100 new mutations per person per generation. I believe that's based on trio studies.
Yeah, I was mostly just curious because there are potentially infinite problems with the way that Jensen did his methodology.
Right, and we can go into that, but that's a totally different topic.
That's why I was just checking. It's up to you. We can talk about genetic entropy, or we can talk about the LMN haplogroups. Whatever you want to talk about, I'm good to talk about it. My closing thoughts are, I think that you got to look to the current literature, and I think that it was handily proven exclusively with math that there is a 25% cap.
We are coming. Hold on one sec. We are coming to the closing of the open discussion pretty quick here. So let's see. We had standing start, and so if you want to do one quick rebuttal, hold on one second.
Discussion-wise,
it's been about 50 minutes.
Well, can we bump it to 60 minutes so we can touch on just a couple more things here, or it's up to you? I know it's getting late for Erica.
I don't have a preference. It's up to you guys. I can stay up 10 minutes later.
That 50 minutes went by fast, Erica. I think we could go at this all night.
I think what we can do is, if we want to, how about we do this? Instead of an extra 10 or 15 minutes, if we want to just Standing for Truth, we can give you another round to kind of make your points, and then we'll give Erica one more round to make her points. And each of those being just a few minutes.
That's fine.
Okay. Should we do a couple more minutes of back and forth, and then, because that kind of sounds like it would be like almost like a concluding statement if we're just going to go a few minutes uninterrupted. I mean, whatever you guys think is best.
Yeah, so that's right. You had heard right. So we'll just have it be like a few minutes kind of making your points, and then we'll give
Sneaky
Erica a chance to make her few points, and then we'll wrap it up.
Okay, who wants to start then their few minutes final points?
As I just said, given that you were about to start, like, you can now start, and then
Okay. So I think since what we were kind of heading into there is genetic entropy, you can answer the question as to, what type of selection is going to be able to filter out all the bad mutations, because the major issue is the fact that selection, it can amplify the best beneficial mutations and it can get rid of the worst detrimental mutations, but it's pretty well powerless to the nearly neutral mutations. Those are invisible to natural selection. I mean, you can think of it this way, Erica. I mean, we've got a genome of 3 billion letters. 3 billion are passed on from mom, 3 billion passed on from dad. But if you take out one letter, say change one letter randomly, are you going to have a huge fitness effect? No, it's going to have a tiny fitness effect. It's kind of like rust on a car. You can't see each rust event, but it's continuous and destructive. Or like a single spelling mistake in a book the size of an encyclopedia, right? Make one single spelling mistake. It's not going to have a huge overall effect, but the accumulation of them over time, yeah, that's going to destroy the message and the book. But why can't natural selection filter out all the bad mutations, Erica? There's two reasons here that you'll have to address. There are too many bad mutations continuously pouring into the population, and most bad mutations are too subtle, as I talked about earlier, to be selectively removed. And we see genetic entropy in real time. Alzheimer's disease, dementia, autism clearly has a genetic component to it. Asthma, autoimmune diseases, cancers, 1 in 3 now, immunological disorders, diseases. I mean, there is a pandemic. It's pretty well entropic degeneration on all levels, and the 100 mutations per person per generation is a fact. And you're going to have to address how that accumulation of mutations is going to be rid of in our genetics, because I mean that's what we see. We see genes are going down and not up. And if fish to fisherman is true, I mean, you're going to have to explain the major problem, which is a net gain versus a net loss of information. So, I mean, if you want to kind of address those points and give me your thoughts on genetic entropy and what your best argument is against genetic entropy, I guess that would be a good way to proceed.
Okay. So genetic entropy does not happen. The ecological term, again, is error catastrophe. And the reason why it doesn't happen, even though ecologists who have no affiliation with creation, evolution, anything along those lines, have tried to induce error catastrophe and have failed time and time again. They've done it with E. coli, they've done it with phages. It doesn't work. And the reason they do it with those is because they accumulate mutations way faster than sort of macroscopic life. So they conceive deleterious mutations are going to build up in a rather short amount of time. And here's the reason why they don't. Near neutral mutations, if they're near neutral and sort of beneficial, then they're going to be slightly selected for. Selection isn't an all or nothing thing, right? Let's say you have a near neutral mutation that is sort of a negative, right? It might not kill you, but it might decrease your fitness, because if it is a negative trait, right? If it is a bad mutation, by definition, it's decreasing your fitness. So you're going to reproduce less than your peers, and whatever offspring that you do manage to produce will reproduce less than their peers, and they will continue to pass on that sort of slightly bad mutation until eventually it is eradicated from the population by one reason or another. It might simply be missed reproductive opportunity. So if it is even remotely bad, selection acts against it. That's just how it is. If it's neutral, and when I say neutral, I mean truly neutral, they can accumulate all they want. Nothing's going to happen. There's nothing to select against. That's the definition of something that is neutral. There is no good or bad kind of impact to it. Now, what you might have in sort of your best case scenario is if an entire group of organisms all get bad mutations at the same time. And as they build up upon one another, they sort of have I think it's epistatic, sort of an impact on where they build up, and it makes them subsequently worse when they combine with one another. But the problem with that is the definition of genetic entropy and of error catastrophe means that it has to accumulate across generations. Regardless of whether it's epistatic or not, the negative mutation will hit the second, and negative selection against sort of this trait will happen the second fitness is reduced. One way or another, that's happening in one generation. So there is no plausible way to suggest that error catastrophe or genetic entropy can occur, because it is the accumulation of bad mutations across generations that eventually harm fitness. We don't see that. Ecologists have tried understanding error catastrophe, because cancers and things like that aren't error catastrophe. They're similar in ways, but again, it's not cross-generational. So understanding it would be quite helpful for fighting those diseases, but they can't do it because it's not a phenomenon that occurs. Sanford, I believe, claims it happened with H1N1, but I read that paper. Not only is it a bad paper that actually makes several mistakes, but it swaps its definition of error catastrophe around whenever convenient. So essentially, my answer to your question is, there is no such thing as genetic entropy. We don't see it naturally. We don't see it in a lab, even though we've tried. At least the definition that creationists give. So it's not a problem because it's not real.
Gotcha.
That's all I got to say about it.
Thank you very much. And standing for truth, I know that you have another round in the barrel. But just for the sake of
Going too well, James.
We can always do the tiebreaker debate. We can do an end game debate. We should call them that from now on.
That's a good idea.
The trilogy end debate where we have like the final of 3. So we definitely can do that, but I do just want to make sure that Erica gets to sleep on a decent, hopefully at a decent time.
You're
So
sweet, James.
it's
I will
no
say that
problem.
conversation flew by. It was a lot of fun.
It's
Yeah,
also
I had a good time.
it's also because I'm tired. Now, next up, Steven Steen, thanks for your super chat. He says, standing for truth cannot be stopped. He's a beast. SpongeBob. You have a
Well,
fan
that is
out there.
That is far too sweet of Stephen to say. I obviously paid him to say that before the debate. If you could read a couple more super chats, I'm running upstairs for literally 30 seconds, James.
Deal. Steven Steen, that sicko, also says, James is the new Adam sent from God to fix us soy boys' flawed genes and restart the human race without flaw or weak jaws.
Listen, James, I have heard you are an alpha Chad. I've heard that from other people.
That's hilarious. An alpha Chad. I had never heard that before, but I like that. We'll call Steven Alpha Chad from now on. Let's see. Bent Hovend. No relation, it says in the name. Says Erica and Dapper Dino should debate Standing for Truth and Kent Hovend at the same time.
I'd be down for that.
That would be epic.
Fun. Could be fun.
We actually, I forgot to mention. So we do actually, it's coming up quick. We do have, as you see in the bottom right, Kent Hovend will be returning. We got off on the wrong foot one day, and I will take responsibility for it. And long story short, for a while, it looks like we weren't going to have Kent Hovend on anymore. But Mark Drisdale convinced him. So you see Mark in the picture there. He, on the left side, he looks kind of like an action hero star, the bald head and the goatee. He has convinced Hovend to come back on. So that will be a good one. But
you don't want to get on Kent's bad side. He'll ban you from Dinosaur Adventureland.
That's true. So it's also Genesis and Cosmology. A discussion will be going on this coming Monday with Skydive Phil, popular YouTuber, really, really pleasant guy, and also Dr. Jeff Zwierink. So that's going to be a really fun one. That's one that we've had planned on 2 different dates because I don't know what was wrong with me. It's the first time I had ever wondered, like, oh my gosh, really wondered. Is there something wrong with me? Like, because I really thought, so they contacted me, they called me, they're like, James, it's not today. And I was like, oh no, you're right. So long story short, glad you're back Standing for Truth. I could hear
You've only wondered that once. I wonder that every day.
Yes.
About myself.
Well, thank you. I appreciate that. And Standing for Truth, I think I could hear you cackling in the back. You must be back.
I'm back, my friend.
Gotcha. Thanks so much. So
wanted to make myself known.
You betcha. Let's see. Michael Dresden with his normal trollish super chat says, I'll save you all the time. Evolution is a myth. You can respond to that, Erica, or you can not dignify it and ignore it.
I'll respond. You're wrong.
Gotcha.
Yeah.
It's short and pithy. I like it. Speed of sound of gravity. Who says, by the way, he said that he or she said, Standing for Truth, you are afraid to debate them. So they said, Standing for Truth, buck, buck, buguck.
Buck, bagok. Like, you know.
Okay,
yeah, Speed of Sound sends his regards, Standing. He says he wants a rematch, so.
Okay, so, and that's funny, Erica mentions that. So I've had 2 debates with Speed of Sound. So definitely over 2 hours, maybe 2.5 hours of dialogue. We debated the same topic that Eric and I just debated, and a topic specific to chromosome 2 fusion. So I recommend anybody go look at those debates. I suspect he only wants another debate because of something called damage control. He knows he lost, but you know what, Mike? Maybe one day. I'm going after the big dogs now, though.
Gotcha. Very sassy.
That's what they call me.
That's right. Jay Shy, thanks for your super chat, said, what if there were people outside the garden whom evolved, then mated with Adam's descendants, still making Adam and Eve our genealogical ancestor? Cain hints to other people who might kill him. So, Standing for Truth.
Well, I would say that we don't even need to go that far because the Bible tells us we came from just 2 people, Adam and Eve, of course. And if you just look at my opening, I went over the fact that we've got these pieces of DNA, for example, the Y chromosome DNA, the Y chromosome we only get from our fathers. and it's passed on unbroken from father to son. And every once in a while a mistake happens, and every time this happens a new branch in the family tree is formed. So all we have to do is look at all the branches in the world, James. They go back to a single person, not to a chimpanzee, okay? And that's what this debate was, limited ancestry versus universal common ancestry, but to a man who lived just a few thousand years ago. And the same thing goes with mitochondrial leaves. So we can just believe the Bible. And when we start with the Bible, as it's just plainly understood, we can make accurate predictions and retrodictions.
Gotcha. Thank you very
James,
much.
if I can make a recommendation for some reading for that individual, they should check out John Walton's series, because he actually subscribes precisely to that, what the question asker was talking about, sort of like a theistic evolution type thing. I've read some of his work. He's an academic. He seriously knows what he's talking about. I'd give it a look. It's pretty interesting stuff.
Gotcha. Thanks so much. And next up, Stupid Whore Energy, as she likes to call herself. Very sick. She says,
I haven't heard from her in a while.
That's right. She says, new genes can arise from non-coding sequences. That's why orphans are not a problem.
Right. So it's just as I iterated earlier. It's called de novo gene synthesis. And I don't want to beat a dead horse, but just go back and watch the discussion. It is a circular-based argument. It's never been observed that these non-functional, non-coding DNA regions are suddenly co-opted to have an important functional role. But orphan genes in general, they are taxonomically restricted. They are functional, and they do suggest, along with many other lines of evidence, that limited ancestry is true, because there's so many lines of evidence that the evolutionists have a major problem explaining, other than weak rescue devices, in my opinion.
Gotcha. Very sassy. Go ahead.
As a question sort of for standing, what do you make of all the astrological phenomena that we draw ties to despite never having actually laid eyes on them in person, like black hole physics or supernova or novae, I guess, neutron stars, things like that? We just get data, we just get readings, and we formulate entire hypotheses about them that then are confirmed by other data that we receive. So you don't have to be a direct observer of something to know that it happened. You can see the aftermath and get the same idea. That's where a lot of science goes. Anyways, that's just an anecdote.
I agree. I mean, you can have all types of hypotheses, but those hypotheses have to have supporting evidence. The evidence has to be demonstrable. It has to be empirical. So some of the things you just mentioned are demonstrable, and the inferences that are made are based on really tight scientific-based evidence. But when it comes to like the Y chromosome dissimilarity between chimps and humans,
we didn't get to talk about that, and I researched that this time, and I was ready for that. We didn't get to talk about it. Dang,
well, remember,
we didn't get to talk about a lot of stuff.
Well, remember, round 3, Endgame, Avengers style.
Listen, eventually I want an Age of the Earth conversation, okay? Or we can talk about anthropology again. I don't like being out of my element.
I enjoyed the human evolution debate, and now we did one on this topic. So yeah, maybe the next one can be Age of the Earth related, or looking forward to it, put it that way.
Very juicy. Mike Billers, thanks for your super chat. They ask, can Standing for Truth re-explain Erica's explanation for orphan genes and maybe stop using it in the future since it's explained?
Well, I know the answer to orphan genes that is given, because it is a major problem to evolutionist theory, is de novo gene synthesis. I can't remember if that's the argument that Erica used. She I guess, look to endogenous retroviruses, for example, and the functions that are now been found in these so-called ancient viral elements have been co-opted. It just comes down to the fact that, are these anything more than just stories, rescue devices? So I understand the arguments put forth by the evolutionists. I just don't see them as being very convincing.
Gotcha. Thanks very much. Moving quickly, Josiah Hansen, thanks for your super chat, asked Erica, protect your hand when giving such a spanking. Very nice. Gotcha. Excellent. Let's see. Next up, thanks for your super chat from Florida man Edo Hypnotoad Evangelist. Appreciate it.
Oh, blessings on you. Priest.
Gotcha. Thank you very much. Let's see. They're asking, give me 1 second. They asked, why can't creation answer the phylogeny challenge?
I'm guessing that question's for me. Yep. I would say that the phylogeny challenge is fallacious, because it all comes down to, where can we draw the line regarding ancestry? Where can we, where does that phylogeny break down? But the thing is, that entire argument misunderstands biology and genetics, because and theology, because they seem to imagine that God created humans and then all of other biological life, and that there's absolutely no similarity between humans and any other species out there. So based on the biblical model, there's really no reason for us to believe that that's true, as in, there should be no type of hierarchy in the biological world. As in, there's no reason to believe that we shouldn't be more similar to, say, apes than to another species, say a whale, right? So we too predict nested hierarchies in biology, whether it's on an anatomical level, physiological, morphological nested hierarchies. So that's why I always say we need to look to differentiating kinds of evidence to show which explanation, limited ancestry or universal ancestry, is true. And that's where we can get into function. We can get into Y chromosome, mitochondrial DNA, predictions. This is how we're going to truly answer the question of ancestry. So long story short, the phylogeny challenge it's fallacious, and it's based on a misunderstanding of science and theology.
Hey James, can I answer this one too? But just from my side.
You bet.
I think the best question that the question that I have for creationists on this one, of course, it has to do with primates, because I love primates and I am a primate. But frequently creationists lump all the apes into an ape kind. And my question kind of runs along those lines, because frequently a kind is described as things that bring forth, organisms that can bring forth with one another. But chimpanzees obviously can't reproduce with gorillas or orangs, or, well, they can with bonobos, but they're actually very recent divergence. And there's been slightly disturbing, but a bit of sort of theoretical research looking into how reproductively compatible humans would be with chimpanzees. And it's more than it is with gorillas, and it's not actually been considered to be impossible, as terrible as that sounds. It's simply a ring around the egg, the zona pellucida, that sort of gate checks any spermatozoans or sperm in general that try to come in. So reproductive isolation is kind of a bad way of drawing the line, which is why I always ask why and how we draw it. And that's why I don't think they can answer the challenge, because there is no way to draw a line. Genetics is a gradient, and it runs through all living things. And that's why paternity tests work. And that's why, genomic comparisons work.
Well, if I could make just one quick point.
Really,
if you just think about it, Erica, okay, according to our model, I'm just going to throw some, kinds out there, say humans, pigs, rats, ducks, and dogs. They're all land dwelling, okay? Even though they are separate kinds according to our model, since they are all land dwelling and share many similarities with each other, they can share similar nested hierarchies or designs. So where are we going to draw the line between those separate kinds if they already share certain levels of nested hierarchies, morphologically speaking, genetically speaking, physiologically, speaking? So that's the thing.
But that's a fancy way of saying you can't draw the line. That's a fancy way of obfuscating that the line can't be drawn. And if your model is correct, then the line should be able to be drawn, because it is, of all the things in Genesis that are like Genesis 1 that are like super clear, there's quite a bit of emphasis on the kinds. It's repeated over and over and over again. You would think it would be quite obvious.
Well, even though we predict nested hierarchies, evolutionists when they say that there's this perfect nested hierarchy, there's this perfect phylogenetic tree of life, that's not actually true, because anytime there's an inconsistency, Erica, and say a lack of uniqueness in that phylogenetic tree, as you know, it's usually explained away by what? Convergent evolution. So that's a rescue device. There's also something called independent lineage sorting. If you look at different genes in, say, humans or apes or gorillas or orangutans, depending on the gene you're looking, can create a totally different type of phylogenetic tree. So even though you're implying that there's this perfect nested hierarchy of life, that's not true. There's many, many types of inconsistencies I can point to.
Gotcha.
Yeah, but the thing is the inconsistencies are, while present, they're present in every branch of science, from medicine to astronomy to, of course, evolutionary biology. That's the nature of the beast. The question is, how strong and how parsimonious are the lineages that we do have these straight through lines? And the answer is incredibly. I know primates the best. It's undeniable from my position. I mean, but that's just me.
We have to
Well, I mean,
standing for truth. I'm sorry, but just to get through more questions, we have to jump through.
That could have been debate number 3.
Yes. Dapper Dino, thanks for your question. Asking to both, are you familiar with the observed de novo emergence in Drosophila or Saccharomyces?
Yeah, so the fruit flies, for example, read that paper. For one, it's done in the lab. It's also inferred. And what they've done in the lab is they've forced these orphan genes out of the genetics of these fruit flies. So he's trying to say that this is an example of de novo gene synthesis, when all it is an example of a loss of orphan genes, which can happen, obviously. So I want to see an example in real time, not in the lab, of de novo gene synthesis, and not just an example of an orphan gene being lost. I mean, that's just...
How would you even do that, though? How would you see the emergence of a de novo orphan gene not in the lab? You're not going to, I don't know, maybe you follow like a colony of Drosophila around with a net, and you catch them every couple minutes, and then you do some genomic sequencing to find out. I mean, I don't know how you would even go about doing that. It's an impossible experiment.
So are you admitting that it's never been seen?
No, it has in the lab, but you just asked for it outside of the lab, so you can't do it outside.
It hasn't been seen in the lab. I just told you that all that was seen was the loss of orphan genes. That's not the gain of orphan genes, Erica.
Hold on, hold that thought. I'm not sure. Let me pull up this paper. James, pull up the next question, if you would, please.
Yes. Next up, thanks for your super chat, Pax Americana, asking, standing for truth, even early church fathers, such as St. Augustine, knew that Genesis wasn't a literal account of creation. YEC isn't even theologically sound.
Technically, if you look at what the plain reading of the Bible is, if you look at church fathers, quote unquote, throughout history, the majority of them did believe that the geology of the earth was explained by Noah's flood. Ponds come to people evolution was not heard of. But the most important thing is we can just look at a plain reading of scripture, and we can see that God created Adam and Eve, and we can also make predictions and retrodictions. And the science tells us we come from a Y chromosome Adam, a mitochondrial Eve. The human species overall has low genetic diversity, which points directly to Adam and Eve. That's why the evolutionists had to resort to the out of Africa population bottleneck theory. I mean, Erica and I discussed that thoroughly in our previous debate, but a plain reading of Genesis clearly is consistent with empirical scientific data. So
look into the Toba catastrophe, if you would, Standing. That's one of the primary explanations for the bottleneck that there's quite a bit of evidence for.
Right, and I did, and it looks like there's no major consensus on whether or not this Toba volcanic eruption is resulted in the population bottlenecks. There's, I believe, some sides in science that believe there's some inconsistencies with that theory, while others hold to it. So, I mean, that just might be a discussion for another debate.
Yeah, I think that it's best explained with a combination of that and founder effect from populations leaving Africa. But that's just me.
Just to move through as fast as possible. Jay Shy, thanks for your super chat. They said Francis Collins, a Christian, mapped out the human genome and is convinced of universal common descent. Why didn't this lead him to your conclusion? I think that he's asking Standing for Truth that.
So he looked at, actually, can you repeat that real quick?
They said Francis Collins, a Christian, mapped out the human genome and is convinced of universal common descent. Why didn't this lead him to your conclusion, Standing for Truth?
Good question. So Francis Collins, I definitely recommend his book, The Language of God. The major arguments he looks to, for example, you'll see him speak of ancient repetitive elements, the non-functionality of our genome, shared genetic mistakes, for example. But he's had to actually go back on a lot of that because he's one of the scientists who has admitted that ENCODE has revealed that much more of the genome is functional, that the genome has many more functional DNA elements than previously thought. And even Richard Dawkins has had to go back on that. I mean, he's the one who quoted, and I don't have it right in front of me, that over 90% of our genome may as well not even be there, implying that most of it is useless. Yeah, Richard Dawkins, just go knock out that 90% of the genome. So it's just based on an evolutionary bias. But yeah, Francis Collins, I'd recommend reading his book, and all his best evidence for universal common ancestry has been completely overturned, just like the junk DNA paradigm has been overturned.
Gotcha.
Now, that 2017 paper disagrees with you, though, Standing. That one doesn't quite agree that 90% couldn't be knocked out if we're dealing with the 10 to 12 range, which is pretty typical. I'm sending you a paper, Standing. I think this is the one that was being discussed. Go ahead, James. Sorry.
Gotcha. And then, let's see. Kevin Tremblay, our friendly moderator, if you can do me a favor, and we usually only time people out or delete their messages if they are using hate speech. So even if they're abusive, like, please ask them like why they're being abusive towards somebody. But in other words, like, try to discourage them. But we usually try to only delete or ban people if they're engaging in hate speech. Next up, Mr. Archaeopteryx, Standing for Truth, an amoeba genome is 100 times bigger than a human's. Does an amoeba need 100 times more information, or is most of it non-functional?
Well, looking at an amoeba genome is a lot more different than, say, testing the genetics of mammals, humans specifically, which is where we're making the direct predictions. But organisms like amoebas, yeah, they are subject to whole genome duplications. Also redundancy, in a lot of these organisms that do have 2 or 3 or 4 times the amount of genetic content than, say, a human, may just need more redundancy for environmental conditions. So, I mean, epigenetics, environment all play a factor in our assessment of the genomes of all forms of life. So, yeah, amoebas, they are subject to whole genome duplication. So some of it could certainly be non-functional.
Hey,
Gotcha.
James,
Yep.
real fast. For those of you who also want the paper that I just sent, Standing, it covers the orphan genes quite well. There's some excellent sources within it covering orphan gene emergence, which I believe, I didn't get a chance to read the entire thing, but it's sort of alluding to the fact that this has been observed in the lab. Not positive. Yeah. Don't quote me on that. Side note, I also had the chance to briefly look, hold on, pause. I'm pulling it up, look at my email, and I had a message from Call Me Emo, who brings up an excellent point that I wanted to post to Standing very briefly. I think it's if that's okay with you, James. It's very short.
Yes.
Emo, yes, I've seen his argument, and we've rebutted.
Well, hold on, I'll ask you the question because it's quite short. He brings up an excellent point that I didn't even think of, but
so am I debating Emo now too?
Well, I saw when I was sending.
Let me just Google some arguments against what you said. Okay, go ahead. Yeah, what's
listen, I just think it's a good point. It's not my point, so that's by far no, but I did think it was quite good. He brings up the fact that orphan genes, if even within kinds, vary wildly from species to species, which would support de novo origin far more than sort of a taxonomically restrained limited ancestry.
Well, he assumes that those orphan genes are found within kind. So, for example, you can find orphan genes that are taxonomically restricted to humans, gorillas, and chimps. But he's assuming that gorillas and chimps are of the same created kind. That might just indicate that they are 3 separate created kinds. And the paper that you posted on the fruit flies, for example, we have an article in our book, Ram and myself, debunking that. What they did technically was they forced adult males to reproduce with infant females to get rid of their orphan genes. So it's unrealistic in nature and therefore irrelevant to the entire argument. Because like I said earlier,
I hate to say it, but if you think that that's unrealistic in nature, I've got some bad news for you. There is a lot of weird sort of sexual crossings across adult juveniles in various instars with arthropods. Arthropods are extra weird. But yeah, it kind of busts up the point that you made previously, though, that because they're taxonomically restrained, they are the ones that dictate what a kind is or isn't. And that
I'm saying it's a line of evidence that can dictate what's created and what's not. And that can actually dictate to us that humans, chimpanzees, and gorillas are all of separate created kinds. But the thing is, orphan genes are very specific. For example, we have noticed that all taxonomic restrictive orphan genes in, say, felines or the dog family correlate perfectly with what I'm saying. So right here, we've identified a feline kind, a dog kind, a human kind, a chimp kind, and a gorilla kind. I mean,
but that's that you want to talk about unrealistic. You're going to tell me that chimps, gorillas, and orangutans are separate kinds, but that every felid and every dog is within the same kind? That's wild, dude. That is like out of this world. The orangs and gorillas, really gorillas and chimps, diverged incredibly recently, even in evolutionary times.
Hold on one sec. We do standing for truth. It's got to be really short and pithy here. Standing for truth.
Yeah, you can finish up standing. Sorry, I keep inciting things, James. I apologize.
I like interaction during the Q&A. It's fun. It's fun.
Okay, gotcha. Thanks so much. Next up, appreciate your question. Speed of sound of gravity asks, standing for truth, what H3K4ME1 in Tompkins graphs indicate?
Histone methylation signatures. It's indicative of active gene promoters in transcription. He's trying to stump me, but I would recommend going to watch my debate. He's really good in genetics, so I've enjoyed my debates with him, but I got to be honest, he didn't do quite well against me on chromosome 2. We discussed it for an hour. It was a good debate. He's even admitted in one of his hangouts with some evolutionists that he was blindsided by some of my arguments. So, kudos for him to trying, in trying to stump me there. But sorry, speed of sound, you failed, man. Big time.
Next up, appreciate your feistiness. Appreciate your question from Terry James, who says, if creationism, quote unquote, predicted nested hierarchies, would disproving nested hierarchies falsify creationism, or would you take this as evidence against evolution?
Well, like I said, there are inconsistencies with the phylogenetic tree of life. That's why they need to look to convergent evolution. For example, I mean, you can see evolutionists invoking convergent evolution, which shows that this so-called phylogenetic tree of life is not as, it doesn't flow as well as they say it does. Because you can even look at an example that at the top of my head, let's say whales, dolphins, and bats, for example, okay? They're the only mammals that have echolocation systems. And maybe Eric has an answer to this, I'm not sure. But on the basis of gross anatomy, the whales and dolphins, okay, they're classified closer to the cow and other ruminants than to the bats. But on the basis of their echolocation systems, they should belong with the bats. Now, it gets even worse for them because molecular analysis of the protein prestin puts whales and dolphins squarely.
70% of women stated
for anyone in the audience who doesn't know, prestin is a sound sensitive protein and it's essential for hearing in general. So it's just one of the mechanisms, hearing mechanisms of the echolocation system. So this, plus other lines of evidence I can go into, is strong evidence that the phylogenetic tree does not represent objective reality and it should falsify universal common descent. But we do
Short and
predict
pithy
both
here.
models some levels of nested hierarchy. So yeah, that's my answer.
Thank you very much. Sergio Sarabia, thanks for your question. They asked, Erica, how does a random evolution process create symbiotic associations? How does nature, quote unquote, select anything to work with another being in ecosystems?
That is a fantastic question, and I think that is quite intuitive, because you would think that would be somewhat of a complicated process, but symbiosis has been happening from an evolutionary standpoint from the very beginning. I mean, we have the endosymbiote theory, or hypothesis rather, which is that quite a large cell sort of phaged another smaller cell, and that's how you get the mitochondria. But the truth is that it's close, loose associations that then have such a fitness benefit for these two organisms being in close proximity to one another that it actually becomes more beneficial for them to stick together than to stay apart. We've seen this recently in certain species of primate use, for instance, vervet monkeys. They can actually rely on the predator calls of starlings. Now, that has been a recent adaptation in many locations. Now, of course, it's location specific because symbiosis does take a long time, but that fitness benefit of being able to utilize a starling call to know whether a predator is around because they have a better vantage point has popped up somewhat, in the sort of distant past, and it's actually led to some really interesting conservation methods where they teach vervet monkeys how to respond to starling calls, and they actually spend more of their time kind of making their sleeping quarters near starling nests because of it. So that's basal symbiosis, but that's how it happens. It happens with loose associations that through time become cemented, and that's what I got to say about that.
Gotcha. Thanks so much. Appreciate it. And we do have another question from Stupid Whore Energy, who asks, she says, slightly deleterious mutations are responsible for neutral constructive evolution. I think that's for you, Standing for Truth.
Right. I'm glad she brought that up. I mean, that's exactly what I was iterating earlier in the debate. They assume these neutral mutations build up in the genome, and they're almost used as a reservoir for change in adaptation and in forward evolution. So that's based on an assumption. What we now know about the genome, and as Erica and I discussed during the debate, most random changes contradictory to the evolutionists are going to be deleterious. But even if, like I said, Erica, even if the 90% of the genome is perfectly neutral junk DNA, as Stupid Whore Energy is insinuating here, that means, and Erica didn't answer this problem, and I don't think anybody can, that means 10 harmful mutations are still arising in every person, every generation. So even the most fit individuals are still more mutant than their parents. And you can have any kind of selection all you want, and it's only going to amplify the best and get rid of the most detrimental, but we are going to degenerate.
Gotcha. Thank you very much. Just to keep moving, Matra J Disco says, Standing for Truth, where does the SIMP gene come from?
The SIMP gene? If it's a like many genes, for example, if we look at allele frequencies, whether it's rare or common ones, they can arise, say, post-Babel, depending on their frequency in the. I'd have to know exactly what that SIMP gene does.
Gotcha.
I don't know. I'd have to have clarification. Do you know what that gene does, Erica?
New to me.
Again, to reiterate, genetics is not my field. I can speak with a decent amount of authority with primates and with human evolution. Genetics, that's why I had notes.
Yeah, I would say that the majority of our genome is created heterozygosity, but there are some alleles that have, there are some mutations. For example, blue eyes, that's based on a mutation, according to our model, post-Babel, post-flood. But we can determine that based on allele frequencies around the planet. So I'd have to know exactly what that does, if it's a mutation or if it's created. It's one of the two.
Gotcha. Appreciate it. Next up, appreciate your super chat from Jay Shy, who says, Standing for Truth forgets we have recombination.
Yep, recombination occurs, but recombination is no help to forward evolution. I mean, recombination and gene conversion, for example, those are a couple mechanisms that we look to in our created heterozygosity hypothesis. That's actually going to create diversity quickly, especially if the DNA differences are built in from the start. It's kind of like shuffling a deck of cards. I mean, you're going to have diversity in every single generation. That's why two people can technically have millions of children, and every single children are going to be different. No two children are going to be absolutely identical, and that's because they're heterozygous. And like I said in my opening, that would be optimal design. If you, that's all I have to say to that one.
Gotcha. Next up, thanks so much. Sigifredo Sarabia, who asks, Erica, is genome degeneration real? If no, what evidence can you give going against it? If yes, what evidence can you give that deterioration of ape DNA doesn't follow the same degeneration before evolving somehow?
So the problem with this concept of degeneration is that it depends entirely on what this individual is talking about. If this is a reference to degeneration, at least in the form of genetic entropy, the answer is selection always selects if it's not neutral. Even if it's partially bad or partially good, selection still occurs. It just might not occur as quickly. As far as variations in how selection works and other kind of genetic concepts like mutation and things like that, mutation is not constant. It's not constant in a single species through its lineage, and it's not constant further throughout into other species lineages either. Chimps have a 30% higher mutation rate than we do, and orangs and gorillas actually have a 40 to 50% faster mutation rate than us. So accumulations of any kind of mutation are going to vary depending on the species and depending on how far back they diverged. Now, again, I'm not 100% sure what they're asking with regard to genetic degeneration, but if it has anything to do with genetic entropy, there is no way natural selection doesn't select. It's not an entity. It's just literally response stimulus with the environment. There's no, people like to say that evolutionists use natural selection as like a god, but that has nothing to, that there's no, it doesn't follow. It's a non sequitur. Natural selection is simply how an organism interacts with its environment on a genotypic and a phenotypic level, and the changes that occur based on sort of a combination of chance and fitness.
Gotcha. Tioga.
Actually, if I could say something, I promise you. I promise, less than 5 words.
I'll give you, okay, less than 5 words, go.
The problem is, that's 3. Natural selection acts on phenotype and not genotype. And I'll just leave it on that.
Tioga, thanks for your question or super chat. They said, I stan Erica, my biology queen, all in capital letters.
Oh my God, you've made it worth staying up this late. That is a lovely compliment.
Very nice. Subtracted, thanks for your super chat as well. They say, hey, Standing for Truth, if Adam and Eve didn't know they were naked, how was Adam aware that Eve came from his rib? Genesis 2:23, you need to be naked to see your ribs.
I'm going to have to think about that one. Obviously a theological based question, but how did Adam not know that Eve came from his rib? Didn't God put Adam to sleep when he took his rib, made Eve out of it? I don't know. I'd have to think about that one a bit.
Gotcha. Jay Shy, thanks for your super chat. They said X and Y Adam were separated between 30,000 years. So put that in your pipe and smoke it.
I was hoping, I figured we would get into the discussion on mitochondrial Eve and Y chromosome Adam, but based on observed mutation rates, the empirical method, trio studies, for example, we can use pedigree-based studies. Adam and Eve only lived just 6,000 years ago, but evolutionists have to counter that by using unobserved and unrealistic mutation rates. So they don't use the empirical method when it comes to mutation rates. So yeah, realistically, you can look at the studies and look at how those dates are derived, and they're really heavily based on evolutionary assumptions. So
Gotcha. Thanks so much. Almost there. Let's see. Sigifredos Rabia, thanks for your super chat. They said, standing for truth, is a platypus a kind of mammal or bird?
Or bird?
Yes.
Because of the bill, I guess.
I would say platypus defies evolution. I mean, what did the platypus evolve from? It looks to me like the platypus is of its own separately created kind. I'm not sure if there's a universal consensus as to the history and the ancestry of the platypus, but yeah, definitely not a bird kind.
Gotcha. Appreciate it. Next up, appreciate your super chat from Stupid Whore Energy. Strikes again. She says, 2 transposable elements in humans and chimps at the same genomic locus means the original insertion was in the ancestor because there are tens of thousands of possibilities for a TE to insert itself within large genomes.
Right. So these TEs, which as he iterated, stands for transposable elements, but they are an important source of highly regulated, they're biochemically active. They are non-coding elements, but they are important in transcription, factor binding sites, and in non-coding RNAs. So they are pervasive in the genome, but they're also functional. So based on their position and location, that's predicted based on our nested hierarchical expectation based on similarities and dissimilarities. But the fact that they're functional DNA elements, just like the endogenous retroviruses, the ALUs, that's exactly what we've predicted all along.
Gotcha. Appreciate it. Next up, Stupid or Energy. Thanks for your super chat. She said neither Dawkins nor Collins have commented on encoding or encode.
Yeah, ENCODE project. Yeah.
Gotcha.
Well,
yeah,
neither
they
of them
have
are
both.
genetic.
I've got quotes from both of them that have commented on it.
You what? I didn't hear that.
I've got quotes from both Collins and Dawkins that have commented on it. Actually, you've referred to the CRISPR debates I've had many times. I've quoted Collins many times on ENCODE and junk DNA in general, and CRISPR had no objection to it. I mean, I'm not sure where.
I don't remember. I can't comment on it. I don't know.
Gotcha.
Yeah, that's I've got a quote from Richard Dawkins here. He had a book in 2009. What he said was, "It is a remarkable fact that the greater part of the genome might as well not be there for all the difference it makes." Then, after ENCODE, in a debate with, I believe it was Jonathan Sachs is his name, he said regarding ENCODE, he said, "I know there are some creationists who have jumped on it because they think it is awkward for Darwinism. Quite the contrary, of course. It is exactly what a Darwinist would hope for, to find usefulness in a living world." So you see that he's kind of embraced the ENCODE results. So I mean, there's a couple quotes right there for you.
I don't know that that's embracing it. That's embracing the work that they've done, and perhaps being open to the idea that there's more functionality than previously thought, which you're right, there is. But the question is how much, and you gotta, I'm telling you, current literature, you gotta stick with it.
Gotcha. We appreciate it. Next up, folks, thanks so much for all of your questions. It's been a true joy. We are going to wrap up just to let Erica get some sleep as she is in London. And so let's see if I remember right. Huh?
You're more tired than I am.
Yeah, that's probably true.
I'm getting pretty tired too.
I'm impressed. Go ahead. What were you saying?
A good 2-hour debate, Erica.
No, this is child's play. I stay up this late. Listen, I've been writing the same project proposal for months. I have to stay up later. I get nothing done.
Gotcha.
There's something called coffee.
Erica says you're a beta if you're tired already. So thanks so much.
That is, yeah. You're a virgin data. And I don't, what is it? Virgin beta cuck? Is that the phrase?
Very glad. Thanks so much. You're the first person that's ever said that on our channel. We appreciate that.
Oh, good. I'm really glad to hear that. That's my claim to fame.
A lot of triggered people out there right now. I appreciate that. That is good work, Erica.
I live to trigger. That's what I'm here for.
That's right. I like that. That's why we have you on.
Actually, quick question. I just caught a glance in the chat, and somebody was saying that I need to address Erica's LMN. Did you bring up? I don't think we discussed that in the
Yeah.
discussion.
So I mean, there
I don't remember
were.
you bringing
Hold
that
on
up.
a second.
I don't.
Hold
I
on.
think I brought it up, and I
hold on
was
a
like,
second.
let's maybe discuss it. But then I don't think we actually did.
We can do that for a third debate, but it wouldn't be fair for me to jump to that question because there were a lot of other questions before that question, and it technically, I don't know if it was originally put in the form of a question, but so I can't jump ahead of that one. Otherwise, it'd be kind of cheating everybody else who was in line first.
Actually, from what I recall, is during the discussion, we were
not going to talk about it, Standing for Truth. I appreciate it. However, we are not going to. I don't want to just ignore all the questions that came in before that. So, thank you very much. If you'd like, there's always a possibility for a third debate. And like I said, I don't want to, for all the people that asked a question before Jeremy at War, I don't want to just ignore theirs. So, thanks so much, folks, for being here. It's been a true pleasure. Erica, what were you going to say? I think I didn't mean to cut you off. I was happy to cut Standing for Truth off, but
no, I was saying that you were
master, James.
I said standing, go easy on James. He's exhausted.
Standing for Truth, my old arch nemesis. But by the way, do want to mention, folks, very excited. As if you didn't know already, I'd maybe mentioned it before, but in case you hadn't seen, we are pumped for the fact that not too far away, actually, we do have a sweet event that we're excited about. If you enjoy Standing for Truth for some reason, we are pumped about the fact that we are going to have none other than Standing for Truth's father. He is going to be on shortly, and that is actually none other than Kent Hovind, or Daddy, as Standing calls him. So that's you'll see that on the bottom right. That will be the end game of their trilogy of debates.
But James, I could have gone my whole life without hearing that Standing refers to Kent as Daddy.
Yes, he does.
So, James, you made that up.
Okay, I maybe made that up. He doesn't really, but I might start telling people he does. So it's been fun, folks. Both of our speakers are linked in the description, though. So I want you to know, folks, if you've enjoyed listening to them, I forgot to mention this up front. If you've enjoyed and you're like, I want to hear more, well, you can. I've put their links conveniently down below in the description. They're waiting for you right now. Actually, I just noticed, Standing, seriously, this is a coincidence. I haven't put yours in there yet, but I'm putting it in there right now. So do want to let you know, folks, we do want to be open to topics. So feel free, if you happen to have any topics, please let us know, as we do want to have more topics, new formats like tag team, triple threat, other things. Like it does help us a lot if people let us know, like, hey, I like this or I don't like this. So feel free to give us that feedback. And now Standing for Truth is officially, he officially has his link down in the description. And so again, thanks. Those links of both Erica and Standing are down in the description if you've enjoyed and want to hear more of them. And thanks for being here, everybody. Thanks so much for your questions. And most of all, we really appreciate Standing for Truth and Erica being here, staying up late and debating for us. We appreciate you guys being here.
Always a pleasure, James.
Always a blast, James. You know that. I'm looking forward to the next debate.
Always a true joy. So with that, keep sifting out the reasonable from the unreasonable, everybody. We hope you take care and have a great night. We'll see you tomorrow with Tom Jump and Benjamin Blake Speed Watkins debating the definition of atheism. So take care. We'll see you tomorrow.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button, because we are just getting started.